Good morning, everyone. It's absolutely fabulous to have you all here, and I must also welcome the many, many people that are attending the symposium online. That's very special, and I hope they will enjoy it as much as you will. So here we are to celebrate. We're here to celebrate science and science excellence with a wonderful symposium title, From Tree Genomes to Future Forests. And I know, having heard the presentations already, that that story will be very clear as we move through the program. Jerry, specifically to you, we hope that you're going to be able to listen to the presentations, other than your own, of course, and see the incredible impact that that first pivotal study has made on the field of forest and forestry. So please also enjoy. To get us started, I have the privilege of welcoming the chairman of the Marcus Wallenberg Foundation, Mr. Marcus Wallenberg, to say a few words. Marcus, the floor.
Thank you. Good morning. Thank you, Mike, and congratulations again, Jerry. Great to have you here. Great to have everybody here today. And as Mike said, a lot of people online, which is great. We also have, I think, 26 young researchers, which we are very, very happy for, because this has been the aim of the foundation for a long, long time, to try to get even more young researchers engaged in this industry in general. And I also look forward to this morning. I think that, as I said last night, our views can be and should be expanded from the foundation's point of view going forward. As geography expands for us, as the science expands for us. And we spoke about this in a small group yesterday, and Mikael Hannu, who sits there and is trying to keep everything together administratively, he will now have to start working, so that's really great. Good. So very shortly, great to see you, have you all here, and let's the show be start.
Thank you, Marcus. Marcus, thank you so much. I think what you will feel, or at least my perception has been in these presentations, that there's a succession, and I think we've chosen quite well, a succession from the early days up to where we are today. So to get us started in that succession, we have the privilege of having Professor Steve Strauss from Oregon State University. Steve has been working in this field for a very long time. Jerry and Steve know each other well, so there are connections which I'm sure you'll come to see in time. I think, Steve, 40 years of working in tree improvement and forest genetics. Fantastic to have you here. Please come to the stage and share with us.
The slide advance is here. The clicker, am I seeing? Is it right here? Very good, thank you. Well, thank you very much, Mr. Wallenberg. Thank you to the foundation for having me here. And I assume the mic and the technical is going okay? Lovely. It's really an honor to be here to help celebrate Gerald Tuskan's great award. Jerry and I have known each other for about 40 years. That's about the span of our career, thus the 44 plus decades of rapid change. So what I'm going to do today, very simple. I'm going to try to look at some of the highlights of change to give you a sense of really how awesome, how amazing it's been, a little bit of context to understand some of the more technical talks that will come up, and then talk a little bit about the challenges of using this amazing genomic and molecular science out in society for good. So we'll end with that and then come back briefly to talk about our honoree, Dr. Tuskan. So this is from a paper that Wout Berrien, who's here somewhere, and he actually was using BioRender to get this done, but we worked on it together. I think it's a nice overview that'll help you, I think, put everything to come in context. So when we do tree biotech or genetics, generally we're using natural genetic diversity. We may amplify it by making hybrids and putting them in unique environments. And then we're putting them in the field in some structured way so we can see their genetic value versus their phenotypic value, their environmental influences. And that's mostly how we do breeding, right? We see, are the trees growing fast? Are they disease resistant? Do they have good wood properties, and so on and so forth? And then increasingly today, and you'll hear some great talks about this coming up, we can use the genome. We can put tens of thousands of markers throughout the genome and use that as an index. We train that empirically, meaning we go to those same field trials and correlate the genomic information with the phenotypes. So it's not based on knowledge of the genes, it's based on just having so many markers that we can characterize the genome in general. So, and this technology, because of the sequencing revolutions that have happened, computation, that is pretty mature technology. Anybody can do it today. It's really a matter of economics. So that's very exciting. That's the left side of the panel in front of you. On the right side of the panel is what I'm calling functional biotechnology. It starts with knowledge of the genes and the pathways and what they do so we can tweak them in an informed manner. We can make a new kind of gene that's expressed a little differently, put it in. It can come from the same species or a different species, or we can edit the gene, change some bases to make it do something we want it to do. And that, and you see this, this ideal of these things coming together. This is like what biotech companies do. They're doing conventional breeding with genomics, but they're also adding in these particular genes for particular traits. And in forestry, really can't do that yet. It's very, very rare worldwide to be able to have the functional side come in. So we'll talk about this cleavage and what we might do about it as we go. So overview, where have we come in 40 plus years? Obviously, genomic methods and diversity tools. I'll talk about the diversity tools in a little bit of detail. We're just coming online. Prior to this era, we really, apart from quantitative variation in the size of trees and what they looked like, we didn't really know what diversity was. We couldn't measure it. We couldn't quantify it. It was a very, very rough science. And then this thing happened with molecular markers that I'm going to tell you a little bit about in detail. And then that led to many more markers, eventually to genome sequencing. So I'll tell you about that paradigm change in a minute. And then, of course, and I think you'll hear more about this from Jerry and others, we have this sequencing explosion of capability, which is both on the laboratory end and the computational end that Jerry and his team helped pioneer. And then we had this thing I'm calling genome-fueled molecular physiology or functional gene biology. I've also called it in some slides molecular-enabled biotechnology. This is the functional side I showed you earlier, where we're using knowledge, scientific knowledge, to make intentional genetic changes, and tell you a little bit about how exciting that is. So an example of where we were at the first IUFRO meeting on tree biotechnology. I think it was 1984, maybe 1985. Jean-Ray, you know this better than I do. But somewhere way back there at the start of my career and Jerry's career, I was at a meeting in Ohio, and the state of genome analysis was represented by this. These are cut curves. We're taking DNA, we're boiling it to separate the two strands, and then letting it slowly come back together and looking at it. And you see that curve up top. It has this very interesting, strange behavior. And that really tells you that there's highly repetitive DNA in the genome, different fractions of it, they're modeled down below. But that was really the state of genome science. This very coarse, it's almost like the Dark Ages compared to the Renaissance. That's where we were at the start of my career and Jerry's. Then what I'm going to do here is I'm going to talk about now this development of markers so we can really see genetic diversity and use it. And there's a paper that I found by some very well-known population geneticists. You can see their names right there, who looked back. And it really helps put in context how important what their work was. And I have some quotes from that paper. So a 1966 genetic paper was a landmark in the study of genome-wide levels of variability, using gel electrophoresis of enzymes and proteins to study variation. It started in Drosophila, the fruit fly, and they used a sort of random set of loci that hadn't been chosen for any reason. So it was, at the time, sort of as close to a random look at diversity as they could get. And this is what one of those things looks like. These are protein extracts put in an electric field, and the proteins are migrating at different speeds because of differences in charge, differences in their amino acid composition. So it represents genetic variability almost at the gene level. And we can interpret those in terms of gene frequencies and so on and so forth. But going on with that paper, together with a study that interestingly was going on at the same time in humans doing the same thing, they found that they could find a relatively unbiased picture of genetic variability, and there was extensive diversity, much more than anybody thought. And there's great discussion and debate that continues to this day about, is that diversity mostly functional? Is it mostly just mutation, sort of noise, or some mix thereof? A lot of discussion in the field at that point. And then finally, and most interestingly for us, the paper stimulated a large research program that found similar things in every organism pretty much that was studied. And then at the same time, that stimulated a lot of theoretical development in terms of how, whoops, let's see if I broke it. Nope, still seems to be intact. In terms of genetic drift, evolution, phylogenetics, so there's a flowering of these computational software tools allowing us to interpret it. But again, these studies were so widespread, and they were going on in the 70s and 80s when my career started and Jerry started, and we both engaged in these marker studies, that we could look back at thousands, tens of thousands of species, animals, plants, microbes, and look at their levels of diversity and relatedness. And these, of course, this led eventually to genome markers, as you'll hear. So what we wanted is we wanted something closer to the actual genome than those protein variants were giving us. And so don't worry, I'm not going to go through all of these acronyms. Geneticists love these things, by the way. This is how we talk, but I'm going to spare you. This is going to show you that what's happened from over time, over the last 40 years, to go from these protein variants to now being able to have tens of thousands of markers throughout the genome and full genome sequences. So one of them was called RAPID, R-A-P-D, randomly amplified polymorphic DNA. It was a brilliant, I remember seeing the talk from some scientists at DuPont where you randomly amplify parts of the DNA. And I should step back and say, I don't know how many of you in this audience know PCR, polymerase chain reaction, but when I started and when Jerry started, there was no such a thing. Whenever I tell my students we were doing molecular biology without PCR, they gasp because everything was so much harder. But PCR also led to these amazing markers like RAPIDs. And so there was a variety of papers. I'm just going to mention one by our honoree, being able to use these markers to very quickly see if you're part of a clone of Aspen or you're part of you're a seedling cohort, which is very important to their regeneration and demography. And this is what they look like. There's these bands on gels. There's really too much information, but some of them behave nicely. You see the arrows here where you can interpret their genetics. And so work like this happened in many different laboratories, and we got enough of these markers that we could almost saturate genomes with them. And we started thinking, wow, for the first time, we have lots and lots of markers almost throughout genomes. What can we do with them? Can we use them for practical purposes? And so the question, could these markers be useful in tree breeding? And so our honoree, Dr. Tuskan, put together a symposium that had an enormous impact on me. He knew that I was a curmudgeon and tended to be skeptical about all these fancy new technologies. So he said, Steve, why don't you express your doubts? And I did it and wrote a paper about it. And it was fairly influential at the time, but it was based on this sort of old marker technology. And so the conclusions, which are mostly that this stuff won't work and won't be affordable, turned out to be completely wrong because we had such breakthroughs in genomic and computing technology. And you'll hear about that from several of our speakers today who are now using it very productively. But I thank Jerry for bringing me together with a lot of other colleagues to think this through. And I've always noticed he's kind of like Abraham Lincoln in the United States. He welcomes his rivals because he wants to make the best choices. He's not afraid to hear divergent viewpoints. Thank you, Jerry. So, and then this thing I'm calling molecular empowered biotechnology. This is hard to describe, but genomics is not just sequences. There are all these other things. Transcriptomes, meaning the expression of all the genes in an organism, in a cell, at different stages of the life cycle. Metabolomes, the different metabolites that are produced. Epigenomes, methylation and DNA modification, we know is very important. So all of this, and then phenomes, looking at the phenotype, and I think you'll hear a little bit about that from Jerry today. You know, whether it be drones in the field or whether it be hyperspectral imaging in the laboratory or in the field, we can really look into the organism with precision and speed unthinkable previously. And then I don't have time to talk about all of this, but being able to do gene transfer is critical. So modifying agrobacterium, the main gene transfer agent, because that's how, when you see a gene statistically in one of these fancy computer analyses, you really don't know what it's doing with biological certainty until you perturb it in isolation of all the other genes and put it back in an organism. So having transformation is critical, and then also having an organism like a Arabidopsis, which is a little plant that completes its life cycle in about 6 weeks. It was the first genome sequence determined. And when we look at these genomes, they're just A, T's, G's, and C's. To make sense out of it, you need to look at what we know about the model plant, Arabidopsis. So everything we do in this area of genomic-empowered biotechnology goes back to Arabidopsis. And luckily, the world had the wisdom to invest in the Arabidopsis genome very early on, and that helps all of us in all the crops, including trees. So because of all this, we really have an abundance of options. We know lots of the genes involved in stress tolerance, lots of the genes involved in lignin modification. The question is really where to start. There's so many opportunities, how to make choices, how to deploy them in the field, and then as we're here, will society let you deploy them? What techniques are acceptable and what are not? So let's move into the second part of this talk, which is really about some of the challenges. So we have extraordinary genetic diversity in forest trees. In fact, those allozyme surveys I mentioned earlier showed that trees are among the most diverse organisms on Earth, bar none. It's extraordinary levels of diversity. And so one of the things that we can do, and Jerry helped pioneer, is have extremely dense markers that can allow us to really, better than about anywhere else, recognize which genes are doing the action. So very powerful. Nonetheless, and it may seem a little paradoxical, when we dissect traits, we tend to see that the major traits that are important in forestry, the complicated traits like yield and wood density, are controlled by numerous genes that have small effect. So Dario is going to talk about that a little bit in the lecture he's going to give this morning online. And that means this powerful technique, there you go, another acronym, sorry about that, GWAS, Genome-Wide Association Studies. That's when you read in the newspaper about a new gene for a disease, a hereditary disease in humans, it's usually isolated through that method, scanning genomes in tens of thousands of people to look for the sources. Well, our experiments tend not to be that big. We can't afford the gigantic budgets you have in biomedical science. And the ones we've done, pretty much for complicated traits, suggest lots and lots of genes of small effect, and you'll see that from Dario. So it's very hard to use the findings from GWAS directly to accelerate tree breeding. So the genes of most importance, if we want to have genes that have big effects, they need to come from other places, not from GWAS. They can be new modes of action from other species, and that can be very, very powerful. And of course, that's had a revolutionary impact on agricultural biotechnology. Or they'll come from these function-based molecular empowered biotechnology studies. And now GWAS can really be helpful here. It may not be the individual genes, but seeing the pattern of what genes are important tell us what pathways are important, and then we can tweak the pathway. I know this is getting into the weeds a little bit. So I just want to make it clear that GWAS is not a useless technique. It's a very valuable technique. But the inferences are not so much about the individual genes, but about the biological findings and how you use it to inform biotech. So fortunately, and you'll hear about this from our speakers, including from Jerry coming up shortly, there's a plethora of findings and exciting things happening from this genome-empowered functional biotechnology. So they come from many, many different places. I've just named a few of them. This is putting it all together to figure out really how organisms function and how we can tweak them. They involve every trait of interest, of commercial interest: growth enhancement, wood quality, pest and stress resistance. And the real problem is that not that we have a shortage of ideas and actual plants even in greenhouses looking attractive, it's really field verification. And you all may know here that plant breeders don't breed on a computer or in a greenhouse, because the performance of plants in the real world is different than the performance of plants in cultured, highly controlled environments. So really it's based on what happens in the field over many years and many different environments. So we really need to be doing that to use this information. The problem is that it's hard. So why do we have this division between, I'd say, genome marker-based breeding and biotech and the functional side? So one thing is the gene function-informed transformation and editing so far tends to focus on simpler traits. It really needs to. And the amazingly powerful work in lignin modification in poplar, and I think John and Wood are here, the Marcus Wallenberg winners last year, is an example of how beautiful this can be. But the simpler traits and lignin is very much a subset of the problems we have in forestry, not the top priority traits. And as far as I know, and please correct me if I'm wrong, these have not yet been integrated into breeding programs. We have some lovely field trials that are very encouraging. But this is one of the best examples that it can work if we get the other stuff right. One thing that's close to my heart in terms of the research in my lab now is transformation as an obstacle. It's very difficult to put the genes in and modify them or edit them at a scale relevant to breeding, which involves many, many genotypes, always changing, always moving forward. And then there's the social side, regulations that restrict where you can grow at all, and then things like green certification that are throughout forestry that say if you use those techniques, you can't have our stamp on your products. We'll decertify the entire company, in fact. So we have these two major social issues. So the world is slowly moving toward relaxing some of them, and I guess it's a hot discussion point here in Europe, but it's completely inadequate to the opportunity, and it's not clear it's really going to make a difference. But there is a little bit of light, and you have to take what you can get. So, and I see I'm running a little low on time, so I'm going to move a little faster here. So this is something close to my heart. So over my career, I've worked with a number of colleagues to write about this constraint, these constraints of regulation and market barriers and ways to get around them that are based on science. So, and this slide, I didn't animate it because I didn't expect to have time to talk about it, but I'd love to. This is great stuff to talk about over a beer, by the way. You know, why do we have this split? Why do we have scientists saying one thing and certain scientists versus other scientists? And you can just read down, there's a lot of very fundamental issues that are underpinning this. So it's hard. It's a hard issue. There's not going to be one answer. So finally, I want to end by coming back to Dr. Tuskan here, our honoree. Like myself, he's been very involved in every step of this evolution over the years, trying these new techniques and so forth, and publishing excellent science. And then something happened. Something wonderful happened. And you'll see the public face of it in a minute. Before I did, Jerry saw the power of genomics and what it would do to biology. So he used his platform at the Department of Energy to rally lots and lots of scientists to sort of start paying attention to genomics, and I think he'll talk about that. And there have been just untold scientific insights, including biotech insights as a result of that. And so to me, it's the genome paper he published that marks the big transition. So it really, so this is the paper published in Science in 2006. It's got more than 100 authors. It's been cited over 5,000 times. It is a big deal in all of biology. And I was actually a peer reviewer. I've told Jerry that. At the time, I didn't let him know. I was a good anonymous peer reviewer. But I'd never read a paper like this before where you had such an interdisciplinary effort, computational scientists, all kinds of physiological scientists coming together to do this genome and then interpret this genome. So it's very, very exciting and really has been part of this revolution we're living through that's empowering this functional biotechnology. So to finish up, obviously no one's going to argue with this. It's amazing where we've come in 40 years, and it's going strong. All the things that promised are happening, providing new opportunities for biotech. But we have this cleavage of what we can do in breeding and what we can do in functional biology as a result of the polygenic nature of traits, transformation limitations, and strong social obstacles. So there's no question either that this guy over here, your honoree, has been a leader at every step of the way. So it's a real pleasure to celebrate you today, Jerry. Thank you very much.
Great.
Thanks,
Steve.
I take this off?
I beg your pardon?
Do I take this off?
You have to ask the technologist here. Thank you so much for a great introduction. Really what we needed as an introduction. Just so that you know, I'm holding a lot of pieces of paper. So I spent a little bit of time yesterday talking to some of the younger scientists here, and I collected a set of questions. And I had a difficult one for Steve, but I'm not going to ask you because I do think we need to move on. But if we have a little time later on, I'm going to select some of these questions. Some quite complex and some a little easier, so I'll have to be selective. We have an incredible honor and pleasure to be able to listen to Jerry. Jerry, last night, most of us listened to you. I was amazed at what you said. I was expecting something more like what you'll probably say, well, I know you'll say here. You were poetic. You talked about the tree and the beauty of the tree. It was really like poetry to me, and it was absolutely fantastic, and thank you for that. We're going to get something different now. So, Jerry, thank you very much. The floor is yours.
It's my extreme pleasure to be here and to look out into the audience and see such successful and well-qualified scientists, both early and late career. And so it's my honor to present this information. I'm going to try to cover the progress we've made using GWAS and Poplar genomics in identifying key genes associated with important economic traits, and then how we're trying to move those into the market and how we hope that they'll impact the future of biology. We're standing here at the beginning of an incredible next 50 years. I wish I would be around for the next 50 years. Biology is going to change the way we live, the way we manufacture, the way we heal ourselves. And you all are really going to drive that forward. And so the next 50 years is going to be known as the age of biology. We humans do the easy things first. It's just our nature. And so the first easy thing we did was physics, and the second was chemistry. The more challenging science is biology. Biology is more complicated than physics, more complicated than chemistry. I have a chemist sitting in the audience here who's shaking his head. John, chemistry is great and very, very important. But biology is more complicated because it infuses all of those things. And we use genomics to try to tease apart what is controlling the way we grow, the way a tree performs under stress. And I hope to show you some examples of that. I'm going to cover the sequencing assembly of the Poplar genome, creating the genome-wide association study that Steve so nicely introduced, some of the discoveries, and a look to the future. So, Poplar's earliest progenitor occurred 470 million years ago, when the first aquatic plant moved out of the water and onto the terrestrial ecosystem. This was a very simple plant. It contained all the future opportunities that we see now in poplar or Norway spruce or Scots pine. It didn't have all the anatomies or features that we see in trees today. It did have photosynthesis, that evolved earlier, but the environment in which this first progenitor evolved was very different than the environment we live in today. And in fact, that event, that first terrestrial plant, which led to all land plants, may have happened right here on the spot we're standing. 470 million years ago, Sweden was in a subcontinent called Baltica. And Baltica was at the equator. And that was the proper environment for where the evolution of land plants may have occurred. Now, this first land plant didn't have roots, didn't have complex cell walls, it didn't have a vascular system, but it succeeded. And it had a toehold on land, which ultimately led to trees. Poplar entered the human record with the Romans. The Romans recognized that you could take a dormant stick of poplar and then drive it in the ground, and it would root itself, and it would then grow from that unrooted stick. And they distributed these dormant cuttings to the populace, to the people. And that's where the genus comes from. It's the people's tree. That's what it's known as. And so the Romans used it to line their boulevards and to create urban environments that were attractive to people. And so Napoleon then adopted that same strategy, although his intent was to march his armies into Prussia and conquer Europe. And in the winter, his armies got bogged down in the snow because they couldn't see the road. And so he used this technique of dormant cuttings to line the streets so his armies could move in the winter. This environment led to many sketches. That's a Van Gogh sketch. And so we can see those poplar-lined boulevards even today. To be more centric to the U.S., Lewis and Clark, who explored the western U.S., used cottonwood to create canoes that allowed them to navigate the rivers. And then, of course, famous battle that led to the United States having Texas as part of its statehood. Alamo in Spanish means poplar. And there was a large cottonwood tree planted in the middle of the mission that then led to the defeat of the Texans by the Mexican army. We began looking very early at using poplar to inform biology. And Tongming Yin here was a postdoc in my lab. He's now the vice president of research for Nanjing Forestry University. And we used microsatellite markers, SSRs, that Steve highlighted, to identify chromosome 19 as the chromosome that contained the genes that determined gender. Poplar's dioecious, meaning there's male trees and there are female trees. And in a breeding program, what we found was that the vast majority of the superior genotypes were male. And if every one of your selected genotypes were male, there was no future generation. So we wanted to identify early markers, so when you're selecting a tree that has not become reproductively mature yet, you could get a balance of males and females. So that was the practical application of this. And we were able to do that very early on without the genome. We could identify markers that would then allow breeders to say, I want 50% females and 50% males in my breeding population. Well, just about that time, the ability to sequence genomes came about. And collaboratively, we worked with many, many, many people around the world, 100 co-authors on that paper. And the folks in Umeå Plant Sciences were key to helping us bring that together. Stefan, Ulve, and Rishi all contributed to gene annotation. At that time, Umeå had the world's best expression or RNA library, RNA being the messenger of information in the genome. And so they were willing to take that unique resource and fold it together with the sequencing to allow us to annotate or to identify genes in the genome. So very critical. At JGI, I worked with Jeremy and Dan, and we began pulling together the poplar genome. This was the first genome, plant genome, to ever be sequenced, assembled, and annotated using shotgun sequencing. Everything before that, humans and Arabidopsis, were done by bacterial artificial chromosomes, back tiling. So you would get a piece of DNA and you just build this tiling alignment. And it took years and years and years to do that. But Craig Ventnor at the Ventnor Institution said, why don't we just chop up the whole genome and then sequence the little pieces and then use computational sciences to rebuild the chromosomes. This had never been done in plants. So we proposed it. The US Department of Energy agreed to do it. And so we were the first to ever use shotgun sequencing. That's the technique that's now been used in every other genome up until today, where you have long read sequencing. We used genetic maps that you saw on the previous slide to build these chromosome assemblies. And from that, we were able to depict the evolutionary history of how did the genome come into being. And it turns out there was a whole genome duplication called the salicoid duplication depicted in that complicated cartoon you see behind there. It was we had, let me say, when we did the shotgun sequencing, we had about 150 pieces of the genome. And we didn't know how they all related to each other. So I spent weeks and weeks trying to put that together computationally and got very frustrated. Sat literally for just a few minutes, rendered my thoughts, asked for help, and all of a sudden that entire genome revealed itself to me. So inspiration or divine intervention, I don't know what it was, but there was this moment where the genome became clear, and we published that. It was also, we knew here, comparing the Arabidopsis duplication with the salicoid duplication, that the molecular clock in poplar is ticking slower than it is in humans and than it is in Arabidopsis. So we think of DNA mutations accumulating at a constant rate, and you can use that accumulation of mutations to set an evolutionary timeline. How long ago did two species diverge? And it turns out that poplar's clock is ticking at a different rate. And the reason that is that you have this long-lived perennial plant that has the ability, when a branch breaks off and it floats down the river and embeds in sand, to germinate and grow and mature. So individual genotypes, not individual trees, individual genotypes can live for thousands of years, 10,000 years, 15,000 years. And those ancient genotypes are casting those ancient gametes into the gene pool constantly. And so every time that event was successful, the molecular clock shortened. And so poplar looks like it's about 1/3 the age that it is evolutionarily. And that's important because it tells us that we have the opportunity to take advantage of that molecular diversity in our breeding programs. So it accumulates this diversity outside of natural selection. And then that diversity is available to us to modify lignin or to find genes for disease resistance. And that's what we did. We began to look for variation in individual markers associated with DNA. Well, Jeremy and Dan and I continued to sequence genomes. We sequenced foxtail millet, brachypodium, kalanchoe, and then ultimately eucalyptus, and you'll hear more about that from Dario later on. Steve DeFazio entered the scene at that point. We had this rich genomic resource, but we wanted to create a genome-wide association population. And Steve mentioned that this is done in humans quite a bit, to link traits to genes. So to do this, we collected 1,500 genotypes along the western coast of North America and resequenced all of those. So now we don't just have a genotype, we have 1,500 genotypes. And then because of this propagation system where you can take a dormant stick, pop it in the ground, we clonally replicated that population 4 times in alternate environments. So now you can begin teasing apart, does the gene express itself only in wet environments, or is the gene expressed in all environments? And that's the power of common garden and association studies. We developed a SNP library. Steve and I began working with isozymes. You'd have 24 markers, you might have 50 markers. We moved to rapids where we had hundreds of markers, but with SNPs, single nucleotide polymorphisms, we had 65 million markers. So now we've saturated the genome. Every 17 base pair across 480 million bases has a variable marker. And this is unprecedented in biology. It's better than the human mapping population. It's better than the corn or soybean mapping population, because now we can look at individual SNPs, those DNA variants, and link them to protein structure. A single change in a nucleotide, ATGC, can lead to an amino acid substitution that then can lead to a phenotype that's valuable to foresters deploying this ultimately in the field. From this, we associated every phenotype we could measure and found 35 genes that went into invention disclosures. There were 16 patents issued out of a collective work over the last 20 years, and 6 of those are in licensing agreements, and I'll tell you a little bit about those. So I had the pleasure of working with Tim Czaplinski, who is a plant biochemist, physiologist, and he has developed the world's leading library of metabolites. Metabolites are small compounds, secondary metabolites, that have impacts on plant performance. We heard yesterday about aspirin, salicylic acid. That's a metabolite. And so poplar is rich in metabolites, and those metabolites affect a plant's ability to withstand drought tolerance. So if you build up metabolites, the leaves can withstand osmotic stress and are maintained on the plant. And so Tim, in his profiling of thousands and thousands and thousands of poplar samples, linked his phenotypic information with our GWAS information, and we've been able to link metabolites to drought tolerance to the genome. And so we now know genes that control that attribute. And Tim's here in the audience. Now enter the picture with Jay and Wellington, and we really hit the ground running here. We took the common garden GWAS population and identified genes that controlled carbon flux in the lignin pathway. There's a gene called EPSP synthase. It's the 6th enzymatic step in the shikimate pathway. 5,000 publications have been issued on this gene. It's the target of Roundup. So when you spray your plant, Roundup interferes with the function of EPSP synthase. And so nobody had ever noticed that there's a small DNA co-option, a helix-turn-helix piece of DNA that came from a bacteria that evolutionarily poplar obtained, and it entered into a regulatory function. So EPSP synthase regulates the flow of carbon away from the flavonoid pathway and into lignin. And so we could begin modifying lignin based on the expression of that gene. Mycorrhizal fungi have beneficial effects or beneficial impacts on plant growth. Poplar has a relationship with Leucaria. This fungus forms an intimate association with the roots of poplar in the lab, but also out in the field. And we wanted to understand the genes that control that. It turns out that there's a single gene called the lectin receptor kinase, which regulates downstream gene functioning, that in the presence of one molecular form, one allele, you could get colonization. If there was an alternate protein form, no colonization would occur. We identified. Steve, can I drink out of your glass? I've been drinking out of your glass for 40 years, so that explains a lot. We cloned that gene and put it in Arabidopsis. Now, Arabidopsis is this 30-day herbaceous plant that completes its life cycle in 30 days. There are no known mycorrhizal associates in Arabidopsis. This single gene, when cloned into Arabidopsis, you could see the fungal hyphae here in wild type. The fungus didn't even recognize Arabidopsis. It grew over the surface of the roots. But with this one gene overexpressed, it began to form a mantle. And then it developed a Hartig net. And this Hartig net is a combination of fungal hyphae in green, intercalating between poplar cells and exchanging nutrients. And so by introducing just a single gene from poplar into Arabidopsis, we changed the entire physiology and behavior, which tells us now when we inoculate field sites in forestry with mycorrhizal inoculum, that mycorrhizal inoculum disappears because the local fauna and flora outcompete this. But by tuning the host with the fungus, we can now ensure that these field inoculations can be successful. And this work was also contributed by Jesse and Jay. Jay's here somewhere in the room, has had a wonderful impact on this whole field. We continued to work, Jay and Wellington, and we wanted to look at the difference, what genes control rough bark versus smooth bark in poplar. If you have smooth bark, you can chip the tree and put it in an anaerobic digester, and it performs well. But if you whole tree harvest and chip rough bark, the bark is a natural antibacterial barrier in nature. And so that ends up killing your bacterial bioreactor. So we wanted to find the gene that favored this and disfavored that. It turns out that there's a single gene, it's a protein, that causes a hairpin or like Velcro structure. So in rough bark trees, the Velcro held the dead cells next to the dead cells, and that leads to rough bark. But the smooth bark, the protein has a single amino acid substitution that causes the protein to flatten, and therefore neighboring cells slough off. That is wild type in humans. The reason we have smooth skin is that exact same protein, the exact same protein in poplar causes a disease in humans called keratoderma, where the protein has mutated and forms hairpin. So we collectively are not all that different at the DNA level or at the protein level or the physiology level than a poplar tree. And I think that insight is incredibly important. Sometimes we think we're at the apex of evolution. Natural selection has led to us. But in fact, we are just one of many, many, many organisms that have successfully survived over time. Thank you. The LSD was starting to get to me. So, yeah. We just recently found a new protein called Booster. This is recently published. Booster's another gene in poplar that acquired a segment of DNA from a bacteria and a segment of DNA from an ant naturally. This horizontal gene transfer occurred evolutionarily a long time ago. So we talk about genetic engineering and the fear of introducing unusual or unintended consequences. Well, this process of acquiring pieces of DNA from other organisms is occurring as we speak in nature. And so we found this gene, and this gene also has a piece of the chloroplast rubisco, which is a part of the photosynthesis apparatus in all plants. And what this gene does is promote photosynthetic quenching. So when a plant comes out of darkness and the sun rises on the horizon and light impinges on the leaves, photosynthesis initiates. And the rate at which that happens is determined genetically. And the height at which that happens is determined genetically. So the faster it can reach its asymptote, the more productive it is. The more carbon that's fixed, the more carbon that's available for growth or defense. So this gene regulates photosynthetic quenching. If it's present, it happens rapidly. If not, it happens slowly. We cloned this gene out of poplar trichocarpa, and thanks to CJ's work, put it into 717, the genotype that's used in transformation around the world today. And you saw increases in 30 days associated with this gene. We cloned it out of poplar, put it in Arabidopsis, and you see the same effect in Arabidopsis. We've now put it in tobacco, soybean, rice, switchgrass, miscanthus, and it has the same effect in all those organisms. This is being licensed today by Living Carbon, a carbon sequestration company, and by the Gates Foundation to put it into cereal production in Africa. And so it is a single gene that has a profound effect on photosynthesis that we hope ultimately will have economic value out in the field. Let's see. So what you're looking at here is our new phenotyping laboratory, and I know you've built these in Umeå as well. The pinch point on information is no longer genetics or genomics, it's phenotyping. So how can we collect or accumulate massive amounts of information and relate that to performance? So this is the GWAS population, not in a common garden, but now in a greenhouse where we use hyperspectral imaging. We have 5 modalities, 2 hyperspectral cameras, a near IR camera, chlorophyll fluorescence camera, and a thermal camera that happen inside these chambers. And so you can accumulate a terabyte of information in a week's period of time. And that information looks like this. And John, Mirko, and Dan are computational biologists. This is a single genotype on a single day with alternate spectra from the hyperspectral cameras. And now what you're seeing is that although we view a plant as homogeneous, its response to drought occurs everywhere, or its response to nitrogen limitations occur everywhere. That's not true. You see these subtle areas within the plant, and we can get a pixel by pixel assessment of that, where we now know that certain traits, certain behaviors occur at the tissue level, not on the whole plant level. And we can identify the genes that are associated with these spectra, and these spectra can be associated with things like nitrogen use efficiency, water use efficiency, bacterial infection, mycorrhizal association. And in a matter of months, we can run entire populations through the greenhouse and identify genes to trait associations. So I think we're going to see a lot more of this high throughput phenotyping, not only in the greenhouse, but in unmanned aerial vehicles, flying over our forests and characterizing behaviors on broad swaths of land in nature. I talked about cell-level expression. So there's a new technique, single-cell genomics, where you can isolate the nuclei out of a single cell. Steve and I used to have to grind up whole plants, get this homogenized set of disrupted tissue, and then run it on a few markers. Now we can sequence individual cells from meristems, root caps, root hairs, the cortex, the xylem, the endodermis, and the second endodermis. And with that, we can identify not only networks of genes that are expressed at the tissue level or the cell level, but we can find promoters that are associated with that network of expression. And then we can take those promoters, and instead of doing constitutive overexpression of a gene on the whole plant, we can use that tissue level or cell level promoter to only turn the gene on in the tissue at the time we want it turned on. And this will revolutionize how we approach transgenics in the future and CRISPR-Cas9 modifications. With that, we've now sequenced 88 new genotypes, full-length, long-read genotypes, and we created what's called the pan-genome. This is variation at the DNA level amongst alternate individuals in a population. So I told you, or maybe I didn't mention, maybe last night I did, that it was a long time ago. I can't seem to remember. It's the LSD, I'm sure. Poplar has about 45,000 genes shared among all the individuals we've sequenced. But when we did long-read sequencing in Poplar, we found that there's this proportion of the DNA that's unique to individuals. So in fact, Poplar has about 75,000 genes. You have the 45,000 shared among everybody, and then these unique genes that are only found in certain individuals. So we can leverage that information now in what's called structural variants instead of SNP, we're looking at whole chromosome deletions or whole chromosome insertions and identify or associate genes to phenotypes that way. And we use computational AI-driven approaches to do this. And then finally, we need to get better understanding of expression. And what you're looking at here is a technique developed by Chauhan, where you can take any gene that you're interested in. And you don't have to have a transgene. You can use this CRISPR-based system to look at where that gene is expressed. And so no stable transformation involved here. It's based on green fluorescent protein and guide RNAs that bring a GFP protein together. And so what you're seeing here are individual genes and where they're expressed at different ratios. And so we can use this technique to then also verify what we see here at the DNA level. This is the expression level, and this is the genome level. So with that, I want to thank my sponsors. None of this could have been possible without research support. And the Department of Energy in the US has been a wonderful supporter of my programs and the programs of many, many others. DOE founded the Joint Genome Research Institute, funds my current research institute, the Center for Bioenergy Innovation, and I'm in debt to them for their 20 years, 30 years of support. And with that, I want to thank you for your attention and just leave you with this one thought. If you, as early career scientists, surround yourself with brilliant, hardworking people, good things will happen. I've had the benefit and privilege of working with dozens and dozens of hardworking scientists who elevated my science greatly, and I wouldn't be here today without them. As early career scientists, do the same. Find those bright, hardworking people, collaborate with them, and you too one day might be standing here on this stage. So thank you very much. Thanks, man.
Very absolutely fabulous. Thank you. I could tell that neither you nor Steve are microbiologists. Immediately when you took that class, and there's a comment about LSD, and I don't want to say any more than that.
It was a joke.
I thought only about microbes. I'm a microbiologist, as many of you know. We have a minute or two,
Okay.
and as you were talking, I was thinking about the early days of doing PCR with a hot water bath and a cold water bath, which we did. Hard to believe for the young researchers today.
And physically moved it. Yes.
Absolutely. We had a name, a person's name for the machine.
Yes.
But a huge driving force, and it would have come out from the shotgun sequencing, has been machinery. So if you read, which I'm sure you have, Craig Fenter's book, Genome Decoded, it's fascinating. If young people should read this book, he talks so much about technology, the access to the machines. So what do you think about that? When you started, were you thinking, I mean, he was going, I think, he linked up with ABI and got every first machine they built, and they broke, and some crazy stuff.
The technology is just amazing. You know, the first PCR machine that allowed me not to move reagents from one water bath to another over a 24-hour period was the size of a small vehicle, and you had to put that in your lab. Now they're modular, and you can pick them up and move them, and my lab has 20 of them. And so not only the compactness, but the affordability. Same thing on sequencing. Those machines now are affordable, and postdocs in my lab can sequence an entire bacterial genome in a day.
And on the lab bench.
And on the lab bench,
right.
Yeah. So the technology has really evolved, and it will continue to evolve. I think AI and computer-based technology will play a big role in the next 15 years. So all of you that still have the opportunity to educate yourselves, become familiar with computational biology and computer power.
Yeah, it's incredible. Congratulations again.
Thank you.
Wonderful.
It's been
a celebration
pleasure.
of science.
Yeah.
Thank you for getting us started.
Yeah.
Thank you so much. We're going to make a paradigm shift in listening now. We have the incredible privilege of having Jessica Paulfiard here with us. Jessica's a politician and has served on the Parliament of the European Union since 2019, I think, Jessica. So we have the opportunity to hear, and Steve raised this issue, the complicated, incredibly complicated regulatory perspective of what we're doing. Jessica, thank you for being here. Stage is yours.
I prefer this glass, I think. So, distinguished guests and ladies and gentlemen, my feeling is that I want to give Jerry my 20 minutes. I could keep on listening to you. It's such a privilege to be able to listen to you yesterday at dinner and this morning. You have a very, very nice way of putting forward your science and research. I appreciate it very much, and I think that everyone in the room also can agree with me. So it's really an honor to be in the same room and have the possibility to listen to you and to others today. And I think it's important that we highlight and celebrate innovation and the scientific progress, which not only shape the future of forest-based industries, but also are essential to deliver on the green transition and fight climate change. It is with pleasure that I join in recognizing your work, Dr. Tuskan, for the pioneering work that you have done on forest tree genomes, which has paved the way for genome-based tree breeding and enabled substantial progress for the global forest bioeconomy. I think the prize is very well deserved, and congratulations again. As a member of the European Parliament, much of my work is focused on topics related to, among other things, the green transition, bioeconomy, climate change, farming, and forestry. I work to create a Europe that is more open to innovation, supportive of science, and friendly to entrepreneurship. I am convinced that science and innovation are the tools that not only can strengthen Europe's position in the world, but that will also provide the solutions to our common challenges, such as climate change. Currently, I am the lead negotiator on the legislative file on new rules for plants obtained by new genomic techniques, so-called NGTs. As you know, Europe has sadly for a long time held back new gene technologies, such as the Nobel Prize-winning CRISPR-Cas9. These technologies have immense potential to not only strengthen the competitiveness of the European Union, but also unlock new solutions to environmental and social challenges. To be frank, the attitude of the EU has been skeptical for too long. And that is hurting us as a union. Our farmers, foresters, companies and researchers all want to be a part of the green transition, to help reduce emissions, protect biodiversity, and build a strong bioeconomy. But they face increasing demands from society to deliver solutions to climate change and produce more and cheaper food and other bioproducts, but they work under heavy regulation. Regulation that stifles innovation. The goal of the new rules for the entities that I am negotiating is to move in another direction, to open ourselves up to innovation and scientific progress, to enable new solutions for our challenges, to provide relief and new tools for European farmers. This is not just political for me, but also a bit personal. My two sons, Philip and Eric, are first-generation farmers and foresters. In just the last 3 years of running their farm, they have already faced both severe drought and floods. They are in the beginning of those who face climate change first. As a member of parliament, but also as a mother, it really pains me to know that these tools that could ease their pain and for other farmers and foresters, they already exist. But the EU regulations prevent them from being used. We don't have to look back far in time. Just in 2022, Sweden was hit by devastating drought. And we can see this climate change affecting farming and producing all over Europe. The idea that my sons and countless other farmers could have resisted the drought, and every drought before or since, almost sounds like an impossible dream, maybe even a fantasy. But the truth is, the dream is no longer out of reach. There is new technology that can develop exactly such crops, ones that are able to resist drought and floods. With new genomic technique, we can make crops more resilient to climate change and to other challenges, but it's not allowed. Not here in Sweden, not in the European Union. In practice, gene-edited plants are banned from being grown, sold, or even tested outside of laboratories. The most well-known method is the previously mentioned Nobel Prize-winning CRISPR-Cas, a tool that allows scientists to cut and paste genetic sequences with incredible accuracy, something that would have been science fiction just a decade ago and has also been stated here. For plant breeding, this is nothing short of revolutionary. Instead of waiting years or even decades to crossbreed plants for desired traits, researchers can now create new, resilient varieties much more efficiently and much faster. We could make plants more resistant to disease and drought. We could allow farmers to use fewer pesticides and protect harvests and yield. These are award-winning and climate-friendly innovations. If we would use them, it can strengthen our competitiveness, it can make us more resilient to climate change, and we can also help to protect the environment. So why is gene editing of plants still not allowed in Europe? The answer lies in a legal framework that was written for a very different kind of science. In 2018, the European Court of Justice ruled that plants created through gene editing should be regulated in the same way as genetically modified organisms, or GMOs. That means long, expensive, and complex approval processes that, in practice, make it nearly impossible for researchers or farmers to work with gene-edited crops. Whether a plant has been already within its own breeding pool through gene editing or has foreign genes inserted, the law treats them the same. And the result? Years of delay before new crops can reach the field. Costs are so high that only large corporations can afford the process, and an innovation environment where small research groups and startups simply give up. Meanwhile, the United States, the United Kingdom, Japan, and Argentina have already reformed their rules. They distinguish between gene editing and traditional GMOs, focusing on risk, not on the method. The consequences is clear. Europe is falling behind, and we need to adapt. At this moment in time, we are negotiating new legislation for plants produced by certain new techniques with the aim of achieving a modernized and science-based legal framework. I cannot really go into the details about the negotiation, but we are in the process, and I'm really hoping that we will have, as soon as possible, a legal and science-based framework in place. There is a strong will, I would say, to ensure that it becomes possible to use NGTs in Europe. So I still remain positive. With that said, the negotiation is not without challenges, as there are still many voices that are highly critical of these techniques, something I will come back to shortly. At this stage, we are trying to find common ground between the European Parliament, which I represent, and the Council, representing all 27 member states of the European Union. The Draghi report, presented to European Commission in 2024, warned that Europe's competitiveness is at stake, that our continent risks being a museum of good intentions if we do not modernize our regulatory frameworks. The report especially calls for a shift from precaution to innovation, a change in mindset that would allow science to develop responsibly but dynamically. As the report highlights, we stand at a turning point. Europe is experiencing slowing productivity, aging population, and rising global competition. The uncertain security situation in the world, with the Russian invasion of Ukraine, also highlights the importance of preparedness, especially, for example, food security. Competitive, sustainable, and reliable farms are essential to ensuring our food supply in times of peace, but also in times of war. And healthy, thriving forests with high productivity and rich biodiversity are essential for enabling a bioeconomy and sustainable energy supply. If we want to maintain our place in the world, if we want to strengthen European competitiveness and European security, then we need to be open to new technologies and foster innovation. The new rules that we are negotiating for the NGTs in one step in the right direction. It is not a complete revamp of Europe's attitude to genetic technologies. It is a simple concept. Plants that are changed by new genomic technologies in the same way that they could be changed with traditional breeding techniques, or where the same change could occur in nature, should have a faster and easier path to approval. It is safe, it is effective, and supported by science. Last year, an open letter to European Parliament, signed by dozens of Nobel laureates and hundreds of other scientists, urged us to adopt these new rules, precisely because science is so clear and benefits are so obvious. It has immense potential. One report estimates not allowing to use of NGTs could mean that our economy is losing out on over 300 billion euros in economic benefits each year. Innovation is key to the future of Europe. We have had a proud history of research, science, and progress. In our universities, laboratories, and companies, we produce some of the world's most cutting-edge research, but we don't make use of it. This must change. Europe's attitude to science, especially in relation to farming and forestry, must change. Sadly, as the lead negotiator for NGTs, I still encounter a lot of skepticism, even within the walls of the European Parliament itself. Opposition to NGTs and gene editing is often not based on any scientifically justifiable concerns, but, may I say, irrational fear of the unknown. Humans have been breeding plants for thousands of years. Modern bananas or carrots bear very little resemblance to their wild ancestors. Yet most opponents of gene editing would maybe not have an issue with eating a banana. NGTs simply do the same thing as we have done to bananas, but faster and with more precision. The anti-science reflex of many when confronted with genetic technologies is not only harmful for our farmers, foresters, and competitiveness, but also poses a threat to the EU's innovative capacity and can cause immense suffering. Many of you probably are familiar with the example of golden rice. Golden rice is a genetically edited rice that is enriched with the vitamin A. A lack of vitamin A can cause severe health issues such as blindness. Hundreds of thousands of children in less developed countries suffer from a lack of vitamin A. Golden rice was developed as a solution by making sure that a staple food, in this case rice, contains vitamin A. Hundreds of thousands, if not millions, of children could be spared from horrible health issues. This should be a success story. But due to pressure from anti-science organizations, the introduction of golden rice has been limited and in some places completely forbidden, without any scientific proof. The result is that millions of children continue to suffer from malnutrition unnecessarily. This could not be allowed to happen. The point of these examples I give is that our reluctance to use new technologies, and our lack of trust in science and inadequate support for innovation do not just harm us on a single level, but across many. It creates difficulties on a personal level for European farmers, foresters and entrepreneurs, such as for my sons. It harms Europe's competitiveness on a global stage. It makes it more difficult for us to deliver on the green transition and fight climate change. And it can cause human suffering on a global scale, such as the example with the golden rice. The same fear and misinformation as in the case of golden rice are what we also face when trying to modernize the rules for European entities. The scientific consensus is a fact. We have a choice whether to ignore it and live with the consequences, or whether to update our rules and regulations in line with science and make use of the benefits. For me, the choice is clear. We cannot let ourselves be ruled by fear of the unknown or what you call the so-called unnatural. Trust in science is what created our modern society. Trust in progress and innovation is what allowed Europe to be the cradle of the industrial revolution. This is certainly clear here in Sweden, where we can pride ourselves with having some of the most advanced industries in the world, from mining and steel production to software development and pharmaceutical research. That same trust is what will allow Europe to move forward into the future and stay as a world leader. As an example for other regions of what is and can be possible. New rules for NGTs are not just one small part of what needs to be done to increase Europe's competitiveness and to foster more innovation, but it is a very important part. It is not just about making it easier for European farmers and foresters, although that is also a very important aspect. But it's also about the broader picture. Few other topics are so clearly illustrated, the division between those who distrust science and prefer to go with a gut feeling, and those of us who prefer to look onto the facts and listen to the experts and form an informed opinion. I have been in politics for many years. I have negotiated many pieces of legislation. But this time, I really feel that this is extra important that we can reach a good outcome, because Europe is at a turning point. Modern science was born here. We have tremendous knowledge, experience, and talent. We have all the conditions that would enable us to continue being a scientific and economic powerhouse. But that requires us to be open to science. Our regulations must be updated if we want to achieve the climate and biodiversity goals, ensure food and energy security, and strengthen our competitiveness. We must embrace innovation. NGTs are one aspect of the journey that Europe needs to make, and I'm proud to be a part of that journey. by investing in innovation, in science, in our forests and our farmers, and in the courage to innovate. We plant the seeds of a more sustainable, richer, and safer Europe. Thank you.
Jessica, thank you so much. We're incredibly privileged to have people like yourself, to have you doing what you're doing, fighting the fight that we all feel very passionate about. You know, we're terrified about hearing that we're in the post-truth era. What does that mean for a scientist? It's absolutely terrifying. I suppose the question, and I'm sure it's in this list of questions, and there are many to you. Unfortunately, we don't have time for the debate that we really deserve. But, you know, what do we do? And the discussion we had yesterday was, you're a politician, you're elected by people, and I think where we fail, perhaps, is we're publishing beautiful work and really important work in great journals, but we're failing to communicate effectively, at least as my perception, to the people that will elect the people we need to be leading us. How do we do better? One question, and then we'll move on.
How do you do better? Yeah, I think that what is important is really to make also science into reality and how it affects people's everyday life. Like we have now had a discussion within Sweden since the war in Ukraine about self-sufficiency, how to provide food for us in times of war, in times of peace. And I think it's very, very close to your heart and to your everyday life. What will happen the day when we don't have enough food for our citizens? I think that we have not been aware of the crisis, and I think that in Sweden we have had for a very long time peace. So we are not exposed to the danger. So I think that discussion is one of those in security aspects, but also for the possibility to meet climate change. It will affect your everyday life when there will be no food on the shelves in your supermarket.
But do we need to get to that point of no return before we actually do something? I suppose is the challenge.
I think, and we have a common, I think, task to really provide people with information. Right. And I mean, I can think I can have opinions, but you have the science. So I think that is also the way to work forward, because we also have to hold hands in order to enlighten, but put information to the general public, what is really necessary and what is harmful and what is not.
We've been challenged. Thank you so much.
Thank you.
Really appreciate you being with us.
Thank you for having me.
Thank you. We now again change direction a little bit. I hand over to Tahani, who is going to speak with us further on the Young Researchers Program. So thank you very much, Tahani.
Yeah,
Job is yours.
So I'm Tahani Kaldeus, and I'm coordinating the Young Researcher event. And this event is in conjunction with the Prize Awards. And this year we've had the privilege to have 26 international researchers, young researchers, to be selected to attend the 3-day program. And the ambition of the program, or the focus of the program, is for the young researchers to network with each other, but also to network and engage with more experienced and senior researchers, which we think is elevating their own research, and hopefully in a couple of years or decades maybe they will be standing here accepting this prize. And we want to promote that they also are encouraged to continue their research. So that's why we have a quite intense program during these 3 days, where we invite external speakers. We have open roundtable discussion with the Laureate, which is highly appreciated. And we also have a pitch competition, or a 3-minute presentation, where the young researchers pitch their research for each other. And today I'm here to announce the winners of that pitch competition. And after the announcement, we will show you the presentation of the first prize winner. But that's not all, because you will be able to watch all of them, because for the I think past 3 or 4 years, we have uploaded all presentations to the YouTube channel. So after maybe a couple of days, we will have the links on the web page. You can download all of them if you want to. But that's actually not all. So also, we will shortly have a break, and during the break, you will see that there is a poster exhibition going on. The young researchers have their posters here, so please take the opportunity during the break to visit their posters. Okay, so let's continue on with the announcements. So we'll start with the third prize, and that one goes to Gerald Tuskan from Aalto University in Finland. Thank you, congratulations. Yeah, so you can take that. Second prize goes to Joel Kastensalo from Luke, Finland. Congratulations. Yeah. And the first prize winner is Marina Melling from University of British Columbia in Canada. Congratulations. Okay, so now we'll present the pitch.
Like many of you, I love cellulose. And you know what they say, if you love someone, you shouldn't try to change them. So in my work, I'm actually looking for ways to not change cellulose while still advancing its capabilities. This is in direct contrast to common ways we see cellulose in technology. Remember those rapid COVID tests we all took? They rely on a chemically modified form of cellulose called cellulose acetate. Cellulose acetate is used because it links to antibodies, the biomolecules that detect infection. But it isn't compostable. And in fact, the EU now categorizes cellulose acetate as a single-use plastic. This is a shame because native cellulose is compostable, and that's probably my favorite thing about it. I wanted to learn why native cellulose isn't used in biosensing devices, like the millions of single-use COVID tests and pregnancy tests that go to landfill each year. I learned that there's no easy way to link biomolecules like antibodies, enzymes, or DNA probes to native cellulose. So technologies that need biomolecules to stay in one place will turn to modified cellulose or plastics as a solid support. The goal of my work is to link biomolecules to native cellulose, and I was able to do this using biology, not chemistry. I designed a protein that has never existed before that is two hands, one that grabs onto cellulose and one that can grab onto any biomolecule. The cellulose grabbing hand is inspired by natural proteins found in soil bacteria. These proteins evolved to hold tightly onto cellulose to aid the bacteria in decomposing plant matter. The other hand grabs biotin. Biotin is a common tool used in molecular biology that can be attached to any biomolecule using a kit that you can purchase from any major science supplier. I had to figure out how these two hands could come together in a way where they're still functional. So I used computational modeling to evaluate many different designs, and then genetically engineered bacteria to produce the most promising two. I am now performing experiments to understand which protein offers stronger bonds to both cellulose and biotin-tagged biomolecules. Besides allowing for compostable biosensors, I believe my protein can also radically reduce plastic waste from biomanufacturing. The production of many pharmaceuticals and foods relies on immobilized enzymes. The surfaces those enzymes are linked to are discarded to landfill at their end of life and persist there. This happens on the order of hundreds of thousands of tons annually. I believe my linking system can change this footprint since it's entirely compostable and can replace these materials. I was able to preserve what I love most about cellulose while expanding its applications. I hope this work inspires you to also use our gifts from forest without compromising their best qualities. Thank you.
So it's time for a break now, and we want everyone to be back here before 11.
We change direction a little bit in the next presentation. So the next presentation is by Dario Grottapaglia. And you might see that on the screen in a minute. So Dario is very, very well known in the forestry business, particularly in plantation forestry. So we get an industrial perspective to the field of genomics and how that is influencing the future, particularly of planted forests. And I don't want to speak for Dario. Unfortunately, Dario is not with us in person, but well, how do I say that? Because he actually is with us. I know that he's online. He has given us a recorded presentation, so his presentation is recorded, but he is watching. At least he's watching me, because the cameras are there. Maybe he's not watching you. So we're going to play the presentation, and Dario, welcome. Thank you so much for sharing with us, and we will then engage with Dario when the presentation is finished. So over to the technologists.
Good morning. It's a real pleasure to speak once again in this prestigious symposium, this time to honor my good friend and collaborator, Gerald Tuskan, a real icon of forest genomics. And of course, it's also a pleasure to be with so many other good friends today. Too bad I could not be in person this time. Going from tree genomes to future forest, I'll briefly try to show how the new science of genomics and the old science of quantitative genetics have converged to cause a paradigm shift in tree breeding. Genomic selection is becoming the applied decoding of the genome, taking us to the modern breeding of future forests. I met Jerry in 1991 at the famous Gatlinburg Symposium on marker-assisted selection. It was a landmark event that started this long journey toward the use of genomic data to impact breeding. It was in 2004, however, that we started working together on the eucalyptus genome sequencing project under the lead of Zander Meijer. This is when we took this historical picture in front of a giant eucalypt in Tasmania. So, Jerry, congratulations. Thank you very much for all your contribution to forest genomics, especially, of course, the sequencing of the eucalyptus genome. The challenges we face for planted forests at all levels, industrial or small farming, are significant. Much higher demand for wood is predicted, but with less and less land available or viable for plantation due to global climate change, particularly regarding changing rainfall regimens. This is particularly important in the case of eucalypts. Now, as we have limited agency to significantly alter the environment, the challenge is faced through genetics, genomics, and breeding when developing more adapted material. So basically, we're trying to understand how genetics controls the variation in traits like tree growth, stem form, wood properties, and stress tolerance, such as drought, for example. Tree breeding has some specific features and hurdles when compared to crop breeding, and some are particularly relevant. Almost all traits of interest are complex, except for some chemical properties, such as lignin, where important advances have been made with the manipulation of single genes. Breeding cycles are long, complex and long-lasting interactions take place between genes and the environment during the long lifetime of trees. And usually we require large areas and long-term investments, which are more susceptible to changes in market demands and the realities of changing climate. Tree breeding has evolved quite a bit in the last 30 years. We have well-developed methods of flower induction, for example, to allow going from seed to seed in 1 year in the case of eucalypts. We can generate large full seed families, clonally propagate almost all genotypes, and use advanced field trial designs for measuring traits of interest with very good accuracy. And in the case of eucalypts, we can also access the wide interspecific variation to efficiently exploit it in hybrid combination to produce real genetic innovation. In this example, by crossing Eucalyptus globulus with the frost-tolerant Eucalyptus nitens, we produced frost-tolerant trees. However, field testing and selection still require several years, even in fast-growing eucalypts, between the time we generate the cross, going through all the steps, to the time that we actually recommend a clone for commercial plantation. So any indirect, early DNA-based selection method or direct gene manipulation would be a game changer to impact target traits and accelerate breeding cycles. This is where genomics comes in. This, however, depends on our ability to map how the variation in genes and genomes directly contributes to the observed phenotypic variation. The question posed here is, of course, what every geneticist is trying to answer in all organisms, and two approaches have been taken to this end. A more Mendelian approach, based on the idea of finding and manipulating single genes of large effect, and a quantitative one, polygenic, based on the idea that many genes control complex traits. Now, this debate, of course, was resolved many years ago by Ronald Fisher, infinitesimal model of many genes of small effect controlling complex traits. The advent of genomic technologies has profoundly impacted these two approaches by allowing, on one side, faster gene discovery, and on the other side, higher predictive power when genomic data is associated with the quantitative genetic methods. Many gene discovery studies have been published in mainstream forestries by genome-wide association studies, where we have tried to associate DNA markers to complex traits and possibly find the underlying genes. Jerry's team, ourselves, and others made many contributions in this area. However, results have not been promising from the applied standpoint. Traits relevant to productivity are polygenic. Hundreds or possibly thousands of genes and their interactions are involved. Detection of this gene is very challenging unless huge sample sizes are studied. Effect sizes are small, usually less than 1% of the genetic variation, and frequently not well estimated, and their validation is challenging. So from the applied breeding standpoint, we do not expect significant impact for complex productivity traits in the near term. Now, we should not feel alone about this. This has been the rule in the top GWAS studies for human height, for example, a trait with strong genetic control. Now, in this study, more than 5 million individuals were studied. 12,000 gene trait associations were detected, covering 21% of the human genome. So 21% of the human genome participates in the control of human height. But only half of the genetic variation could be accounted for by those 12,000 associations. The rest remain undetectable. So this is probably what we would find for tree height and drought tolerance, for example, if we were able to carry out such a huge experiment in a forest tree. And independent validation of the associations found has been very challenging for many traits and diseases, even in humans, where many studies have been done. Validation rates are usually less than 10%. In trees and plants in general, there is very little independent validation. Jerry's team has made the largest advances in this area in forest trees. Recognizing that the architecture of complex traits is highly polygenic, the convergence of the new genomic technology and time-proven quantitative genetics has now resulted in this new paradigm. For applied breeding, we have moved from trying to discover individual gene to capturing the full aggregate of the genome effect like quantitative genetics has historically done, but now using genomic data. Taking us to the very concept and practice of genomic prediction of complex traits, a field that has experienced a huge development in the last 20 years in animal, crop, and tree breeding. Now, genomic selection follows a very simple concept. You select on a large number of markers to capture the entire genome-wide effect in predictive model, irrespective of understanding the underlying mechanism. So imagine a genome with 5 chromosomes, these black bars, and you have an unknown number of genes, the red squares, of unknown effect. You just use a large number of markers, these gray arrows, to select on, and therefore capturing the effect of all those genes that are controlling the traits of interest. Yes, of course, it's a black box method. I call it the black box of how evolution created all those mechanisms. Now, the practice is also relatively simple. Mathematical models are trained based on field data, here in the parental generation, where trees are phenotyped and genotyped in what we call the training population. Now, this model is then validated in an independent validation population, and once we have a good, accurate predictive model, we can use it to select on the seedlings derived from that generation at early age and carry out what we call the genomic selection cycle. So these genomically selected seedlings are then ranked based on their genomic predicted genetic value, and depending on the interest, you can either clone them and test them in the field for validation, or you can induce flowering quickly and cross them and turn a new breeding cycle in the program. This approach, of course, can radically increase genetic gain per unit time, which is the measure of efficiency of a breeding program. We can increase selection intensity, which is the number of individuals that we can screen. And of course, the more individuals we can screen, the better chances are that we find the best ones. We can increase selection accuracy, which is the correlation between the genomic breeding value and the true breeding value, because we can get closer to the genetics of the individual. And we can reduce breeding cycle length. So by tinkering with those 3 variables, we can increase genetic gain per unit time, increasing the efficiency of a breeding program. To carry out genomic selection, we use SNP markers. Single nucleotide polymorphism is the most common variation in genomes. For eucalypts, we have developed operational SNP platforms used worldwide for the 20 or so eucalyptus species by more than 50 companies or research institutions. Now, we currently genotype at the cost of about $25 per sample. Now, genomic prediction in trees typically show predictive abilities between 50 up to 90%, depending on the trait and the population. In the y-axis here, we have the observed breeding value, and the x-axis, the genomically predicted breeding value, for 15 different traits in eucalypts. Chemical traits such as lignin content and composition, or cellulose, have higher predictive ability because they are relatively simpler traits, while growth and physical fiber traits usually in the range of 50 up to 70%, depending on many other variables. In this particular study that took 7 years to complete, we showed for the first time prospective genomic selection results across generation in a forest tree. We reached high prediction ability and, more importantly, high ranking correlation between what trees we would have selected at harvest age at 6 years, on the left-hand side here, the ranking, compared to what we had genomically selected at age 3 weeks in the nursery on the right-hand side of the graph. So we could get a ranking correlation of 87%, and within the 5% top selected individuals, we could correctly rank 60% of them. Again, we're not alone. In the last years, human geneticists have adopted the same genome-wide prediction approach of complex traits with the so-called polygenic risk score, an estimate of susceptibility to a complex trait or disease. For example, in this particular study, 2.1 million DNA markers were fitted in a model to predict obesity. The accuracy reached here was about 40% in the top decile. So we actually get better accuracies in trees and plants in general, as we have seen before, for a number of reasons that I won't have time to discuss today. But the interesting point here is that in human genetics, the same approach is being used not to select, of course, but to predict the risk. As a genomic breeder, I've used an analogy, which is also a bit of a tease with my fellow molecular biologists. Genomic selection is like investing in index funds. It's cheaper, simpler, humbler, and generally more efficient. Instead of trying to pick single winning stocks or genes, we breeders simply buy the whole market, or better said, the whole genome effect. Now, many financial studies show that this tends to be more efficient on the medium to long term, and we believe this is the same case in tree breeding. In conclusion, these are the advantages and remaining challenges of using genomic selection in tree breeding. We can increase genetic gain by accelerating breeding cycles and especially increasing selection intensity. However, the environment continues to be a moving target, so that predictive models have to be updated regularly with new field data. Cost of genotyping is still an issue, but it's coming down. And of course, a criticism can be made that we're not advancing in any mechanistic gene discovery and understanding, if that's the objective. To deal with the challenge the environment represents, environmics approaches have seen a rapid development in recent years. Just like genomic data allows measuring and predicting the relationship between tested and untested individual trees, environmic data, which is large-scale environmental data, allows doing the same between tested and untested environment. In Brazil, we have done a significant progress in using large-scale environmental and genomic data together to guide the prediction of performance of untested trees in untested environments. Research and implementation of genomic selection programs in tree breeding has been internalized by several eucalyptus forest-based companies worldwide, using the genomic platforms we have built and developed and the approaches we have shown. So a large number of publications have been reported in recent years, and we see a very bright future for the operational use of this technology. I'd like to thank all the collaborators that worked with me on this journey since 2008, actually, when we started working on genomic selection. And thank you very much to all of you for your kind attention today.
Dario. Thank you very much. I was going to say, I can see Dario, but you can see him too. It's great to see you. You look fantastic. Just very sorry, Dario, that you're not with us.
It's okay. It's, next time.
Next time. Next year, you promise us. I've collected a few questions from some of the team here, some of the young people, and I'll throw those at you because you've given us some time to deal with questions. I'm not going to ask for questions from the floor because that'll take too long, although I am tempted. But Dario, I thought you've listened to the symposium through, and what time is it there? It must like 5 a.m. or something.
Oh, it's 7:20.
Oh no, I was feeling sorry for you. Yesterday we were speaking about poor old Dario, but coffee is very important in Brazil, so you you kind of keep yourself awake. But Yeah, cheers. Dario, you've listened to the presentations thus far. I was wondering whether you don't have a comment or two to make, perhaps to Jerry or to Steve, I have.
Jerry and Steve have been my mentors in many ways. So I'm just so happy to be able to have worked with Jerry and also met with Steve and so I mean it's just great talks, great stories, and the glass sharing was also funny. So
yeah, I'm still thinking about those bacteria, but we won't go into that now. So Dario, genomic selection, I mean, you've done tremendous work, you and others, but you're really the leader in terms of eucalyptus plantation forestry. Listening to, for instance, Jerry, so there is a dichotomy, the whole haystack or the gene. What do you feel about that? I want to pivot that particular question towards, for instance, regulation and genetic modification, and CJ's talk that follows, and you know her as well, we'll get into the genetic modification space. But how do you feel about that?
Well, actually, they are extremely complementary. I mean, I think there is a great future for both approaches, especially when they're combined. And I'm sure that the concept of identifying and manipulating single genes of larger effect will become more and more useful in breeding, although the challenges are definitely not small for a number of reasons that I mentioned some of them, but they are totally complementary, especially when we have traits for which we do not have enough variation in our gene pool in the specific species we're working with. So, I mean, for example, in lignin manipulation, there have been just great advances, and I'm sure that this is an area that the prospect of CRISPR, gene editing, multiplex gene editing, has been shown actually, and so I think it's extremely useful and complementary to the way that we're looking at polygenic traits related to productivity and adaptability. So that's my short answer.
So that was a piece of a question that I had on the paper, but there's this whole issue of regulation, and you heard the presentation that we had from Jessica, so European Parliament, relating to regulation. I mean, this is a big issue for us is acceptability of, say, for instance, genetically modified or genetically edited trees. How do you feel about that? I know Brazil is quite far ahead in that particular space, but integrating that into intensive plantation forestry using, for instance, eucalyptus, so a non-native species in a naive environment, I guess.
Yeah. Well, I think, Steve Strauss, he's the expert on this topic. So, it's very difficult to say anything. So I think, again, especially in eucalyptus where we can clonally propagate relatively easily, I think the prospect of being able to use genetic modification, especially now with the gene editing technology, I think are great. Again, the challenge is really having the good target, understanding the mechanism, how to edit whatever we want to edit. And I think the future, as Jerry put it very clearly, I think the future for the next 20, 30, or 50 years are bright. And I have no doubt that acceptance will happen, especially as we face increasing challenges in terms of being able to plant and grow trees in different environments. Here in the tropics, we have upcoming challenges, especially related to water distribution or rain distribution. And so I'm sure that the combination of a more polygenic view of adaptability trait together with traits where we can generate new mutants, let's put it this way, new phenotypes that we're not able to generate by conventional breeding will be key for the future of our planted forests.
Yeah, you talk about the challenges, and as you know, I'm particularly interested in what pests and pathogens are doing. In fact, I think maybe water, but I think that is the single greatest threat to plantation forestry globally. A subject for another time, but I will tell you that I was very interested in one of your slides, emasculating eucalyptus flowers. And you have a really interesting disease on those leaves. So I'm wondering whether you go look at your slide. I am wondering whether you're choosing the best trees, but that also for another time. You've disappeared from my screen, but I can see you at the back. So Dario, you made an interesting point, and Jerry did too, that in many ways the technologies that we utilize and that are being utilized in tree breeding and genomics relating to trees has followed human genetics and human studies. And you gave examples of that. You sort of said, but we're not so bad and we're more or less better than them. But I mean, a question in my mind, for instance, and it also was captured in a point that somebody made in our discussions here, was what are the medical researchers? Firstly, why are the medical researchers ahead of us? Where are they now? What are they doing that we're not doing that we can learn from? And maybe the other way around, what should they be learning from us?
Well, I think, funding, it's very different in many ways. And the buildup of huge databases, like, for example, the UK database, where they just resequenced several million individuals, and they have phenotypic information on clinical data for the last 50 years or sometimes even 100 years. Sweden also has also done some huge developments in this area. So I think we really would need something similar, because we learned that, especially for dealing with complex traits, we need huge sample sizes. And of course, Jerry has done some of the best efforts in this area with poplar. He mentioned the resequencing of 1,500 trees, but we'll probably need more like 15,000 or 1.5 million trees resequenced and phenotyped in the field, different environments. And that's something that we might be able to do in the future.
And you made a very, very important point, maybe a sensitive one to all of us, and that is the issue of money. I oftentimes think about the COVID pandemic, and my physician said there's no vaccine coming, and the reasons. I mean, he showed me Fauci's book, and a year later we had vaccines, and many of us might not have been here today if that hadn't been there. So what happened there, other than the so-called blue sky fundamental research that was done many years before and probably got a lot of criticism, what really happened is they threw the bank at the problem, the money. So I guess we've just got to keep pushing for the funding, keep convincing the industries that there's something to be gained. But it did make me think about, you've done this tremendous work on eucalyptus, and these huge areas. I think I looked the other day, about 35 million hectares of eucalyptus planted in plantations. That's non-native eucalyptus planted in plantations. But there are other trees. So what about acacia, for instance? And there are many, many millions of hectares, not the same as eucalyptus, but approaching that. Why are we not seeing the same drive for eucalyptus pine? For acacia, pines a little bit more complicated. But I'd like your thoughts on that. And then we're going to stop.
Actually, in acacia, there have been great efforts in Indonesia. I've followed that, including some publications, and they are already applying genomic selection as well. They just don't publish a lot because those are companies investing, so that they're not, driven to publication. Of course, in conifers, great efforts, great advances have been made in the US, in Canada, in Sweden, and Harry Wu will talk today, so I'm sure that he will tell us great stories. So I think overall we see a great interest and moving forward into the use of genomic prediction, genomic selection, and breeding program.
Yeah, Dario, we have to end. I hope you stay with us. We see you on a big screen. You look great. No, really, and the presentation worked really well. When people say recorded and online, I think, oh, here we go. but we have fantastic technologists here, so we must be grateful to them too. Thank you so much for joining us. Enjoy your coffee, and we continue.
Thank you
Take
very
care.
much.
Bye.
Thank you very much, all of you.
So, we move on. Our next presentation comes from CJ Tsai from the University of Georgia in Athens, Georgia. I had the special privilege of visiting with CJ last year when I was on sabbatical, and in fact, maybe led to some of the discussions that we'll hear here today. CJ is seriously into genomics research, how trees defend themselves, and we'll have a little bit more after you, CJ, on this whole issue of tree defense, and we'll talk some more. Over to you.
Thank you, Mike. It's a pleasure to be speaking at this symposium on popular genome engineering and its role in the emerging bioeconomy. I will just start by sharing a couple of CRISPR genome editing stories in popular on chemotypes and morphotype engineering, and then quickly introduce two intriguing but underexplored genomic features that would not have been discovered without genome, orphan genes and DNA satellites. Before I go too far, I want to draw your attention, of course, we celebrate the very first popular genome, the very first tree genome. It was wonderful thanks to Jerry's leadership and foresight. It has been foundational to research, including CRISPR. But for transformation, for genetic engineering, the worldwide most commonly used model is 717, coming from INRA's breeding program. Here, again, unit breeding and genetic engineering. It's really easy to transform, and undergrad in the lab can do this, but it's a distant cousin of the trichocarpa. So the sequence variation is huge. I like this cartoon because mining genomic information is a delicate balancing act, like walking the tightrope. But before I dive into CRISPR, I'm sure you all heard about CRISPR, but I will give you a minute's quick introduction. CRISPR reflects the evolutionary ingenuity of bacteria. They sense invading enemies like viruses, plasmids, chop their DNA up into pieces, and then store those DNA pieces into their own genome. You can think of those colored boxes as a memory system, ready to guide and attack when the same invader returns. Purple attack purple, destroy their genome. So this is a survival strategy. We heard from Jessica this morning, many countries still debate GMO and genome editing. Bacteria has been editing their genome for millions of years. If you like yogurt, if you like cheese, you have been eating CRISPR without regulatory approval. But to use CRISPR, it's incredibly simple. You need two things. You need an enzyme called Cas, and then you need a guide RNA. You can think of Cas as a GPS with scissors. But GPS won't work unless you give it an address, and that is the guide RNA. It's just a short 20 nucleotides, 20 letters long. With the two, once they arrive at the target site, DNA got cut up, the host repair the damage, and then in this process, generated a few nucleotide change. But to do that, in practice, you need to know the genes, or better yet, the genome. You need to have a way to deliver those genetic tools into the cell and then regenerate the cell into the whole plant. Steve already talked about lots of challenge there. Not all trees can be regenerated and transformed, but at least we have an incredible tools. So 10 years ago, when we started CRISPR, we have to manually check the guide RNA, the address, to make sure we don't run into the variation between 717 and the trichocarpa. And then what you are seeing here is the world's first gene-edited trees. We target the lignin gene. It worked beautifully. We have specific targeted edit. The lignin reduction is like a flat lines across 30 different events. They all show the beautiful coloration. Easy and simple, right? Only if you get the right address. Because the second gene we target totally failed. We missed 2 SNP circles in the red. So they just told us that the technology is only as good as you know how to use them. So with the lesson learned, we start building a database, comparing the trichocarbon and 717 genome, cataloging all the sequence variations. So then when we design the guide, we can check and not fall into the traps again. This worked really well. When we target a set of regulators, they regulate trichomes. As you can see, we generate mutants. They are totally lacking the hair, targeting a known gene plus their regulator. So you can see the phenotype, but when we map the DNA, it was very confusing because the program was telling us that even our wild type had edited, which was, of course, not the case, right? This was not resolved until we sequenced the 717 genome. Shown on top is the trichocarbon reference, 717 being a hybrid. We have 2 genomes. The middle one looks exactly just like reference. This bottom one from Trimula actually has an extra set of 2 genes, and they complicated the mutation mapping. So having a high quality genome, it's not just important for designing the guide, it's also very important to mapping the mutation so we know exactly what happened in the experiment. And we now have better tools to improve the editing efficiency. So with the mutants, I hope you can appreciate that in the wild type, you see those fuzzy hairs heavily coated with trichomes. So when we have hairless mutants, we simply ask, how does that change the surface chemistry? So with the Hellboy's team, we did a leaf wax analysis, dipping the leaf into the solvent for like 30 seconds to look at the wax. We were surprised to find that the triterpenes were totally gone. That's total up there. Here are breakdown of individual compounds, not only in the leaf wash, also the whole leaf when we crush up the tissue. So this taught us, the mutant taught us that populars, prior to this study, we know populus mutant accumulate phenolics. We were the first to show that populus mutant actually, populus trichome also accumulate terpenoids. So with that, setting the foundation for another DOE-funded project called BioPopular, led by Robin Buell at the University of Georgia. We try to manipulate this triterpene pathway, blocking this branch to direct the flux toward bisabolin, which is a precursor for fragrance, perfume, but it's also a precursor for jet fuel. So the idea is to target specific genes for triterpene. As you can see that we have already mutants with reduced the triterpene level. We already have a number of candidate genes that we know can be used to drive bisabolin expression. Populus does not produce bisabolin, but pines and some additional plants do. And we want to do this in cuticles and trichomes. This is intentional. We don't want to interfere with wood chemistry, the traditional use of wood biomass. So we been working on this, turned out to be harder than we thought, but we now have a set of gene switch that can drive the bisabolin gene expression specifically in trichomes and in cuticles. You can see in tobacco and Arabidopsis, they produce different kind of trichomes than populars, and they all show, especially we are excited about the third one on the right, it drives very specific expression in the cuticle layer and then in the secretory vesicles. So we got a gene switch. We want to do this in the morphotypes that produce a lot of leaves. So we are collaborating with USDA scientists who has been working on fruit trees breeding and genetic engineering for decades. So we translate their knowledge to poplar by targeting a number of architectural genes. And you can see examples on this photo from tall and spindly to short and bushy. You can do breeding. It will take you probably centuries to get this, but single gene, single transformation, we can get diverse the phenotype. And the phenotypes are even more pronounced in the field. This picture was taken by Andrea postdoc 2 months after outplanting this summer. Those are the ideal phenotype to be staking the novel chemistry traits, and there are also opportunity to optimize populars for a diverse applications. So switching gears to the second part of my talk on some hidden genomic gem that I call them. The first one is orphan genes. You heard Jerry talk about this a little bit. Across all plant genome, actually any genome, human genome, bacteria genome, you always find a small percentage of lineage-specific orphan genes not found anywhere, only in the genome, shown in the purple or the red box, about 5 to 10%. So Jerry talked about this example, a popular gene originating from a piece of Chloroplast rubisco, and a piece of bacteria, and a piece of ant to form this fusion now reside in the nucleus. It's only found in some populous Trichocarpa individuals, not all of them. When we overexpress this gene in 717, again, Jerry showed this picture, 717 doesn't have this orphan gene, it boosts the growth like crazy. This isn't the only case. In fact, Wellington and Brooke found 2 other such orphan genes have similarly amazing growth boosting effect. Orphan genes tend to leverage existing pathway to convert those advantages. So that means that they can work across species boundary. And again, Jerry showed this, the same booster gene you put into Arabidopsis. You can see the effect. Then you ask, with so many orphan genes, what can we do? So my lab actually did a reverse. We mine Arabidopsis orphan genes to improve popular. And I love this story because this was the COVID shutdown. Surprising discovery. After 6 months, when we got back to the lab, some tissue culture cups, we expect these, but we saw a few amazing transgenic. They survived the neglect of several months of no subculturing. Media totally dried out. So this set of genes, in popular, when we stop watering 4 days after wild type already droop and suffer irreversible damage, the transgenic is still perky, retain their turgor. The genes were known to be cold tolerant. We show could be drought tolerant, nutrient stress deficient. So this is an example that we can probably engineer multi-stress tolerance. Finally, let me talk about another understudied genomic features, satellite DNA. You heard about microsatellite markers. But satellite DNAs are named because in this case, it's a discovery from the high-quality genome, 147 base pair repeated millions of times, forming an array, drawn in the blue bar. Some of them are even larger than the centromere repeat. The amazing thing is you can actually see them when you do this chromosome squash assay because they're so abundant. What postdoc Ron did is survey all the populous genome available out there, and then we color-code the populous name. You can read it, but we color-code them by the sections. You find them only in the aspens, in the light blue color, except for a tropical aspen only found in this Hainan island, geographically isolated. Why is this significant? Because aspens actually don't get along with any other populace. It has been notoriously difficult to cross aspens with other populace, meaning that many populace programs could not tap into aspen gene pools. So we wonder maybe those satellite, aspen-specific satellite repeat hold the key to breeding barrier. So we decided to do a crazy experiment, edit them out. Of course, this is a proof of concept. We build construct because they are highly, highly similar. So we were able to generate tons of catalyst events, but only 2 or 3 can get to the whole plant regeneration. Probably because we have millions of copies. If you cut so many of them, you could have lost a whole chunk of chromosome. So when we sequence the genome, that's what we see. On the top is the wild type, flat line. The sequencing really kind of distribute evenly. But in the transgenic event, you see those dip. And then in the callus, we have more of those dip. And then zooming in, you can see the Lego mapping correspond to the repeat marked by the blue bars. So the project is still young, but it shows the power of CRISPR to edit not just the genes, but potentially the non-coding mysterious repeats that could inform us about their function. So to wrap up, the first tree genome and those that follow revolutionized tree bioengineering and setting the stage for CRISPR genome editing. We now have the tools to target multiple genes and multiple traits at once. We can target not just genes, but also non-genic region. We can also start thinking about addressing long-standing questions like hybrid incompatibility. Obviously, there are still challenges like studying all of the genes in the genome, transformation bottleneck, regulatory issues that we heard this morning. Nevertheless, genomic science has transformed my own career. I believe it will continue to shape the next generation of scholars. To close, I want to thank you, Jerry, for the leadership and the foresight. They really has positioned us at the forefront of plant biology. So on behalf of my colleagues, my lab, Team BioPopular, and Center for Bioenergy Innovation, thank you and congratulations.
CJ, fabulous. Thank you so much. Those pictures you have of trichomes with that are fluorescing are fantastic. It needs to be maybe of nature or science. That would be good. Just really, really fabulous. Our next presentation, Sanushka Naidu from South Africa, from FABI in South Africa. Great to have you with us, Sanushka. Sanushka is a molecular biologist, and she and her team apply their research to understanding pest and disease problems in plantation forests.
Thank you very much. Ladies and gentlemen, it is such a pleasure for me to be here and to share with you the work that we are doing, really harnessing the power of genomics to enhance tree resistance. So climate change may pose two scenarios. The first scenario is that there'll be a negative effect on pathogens and pests. This means that we will have, shorter breeding cycles, maybe less insects. Maybe we're going to have unfavorable conditions and we're not going to have enough pathogens around, which would be great. That means that we would have increased productivity in our plantations. It means that we would have better wood products and, of course, economic gain. The other scenario is that we can have a compounding problem. We would have biotic stress, which is pathogens, insects, and we would have these abiotic stresses, such as drought, heat stress, cold stress. And this is going to render our trees even more susceptible. This is going to lead to damage of our trees, and then we're going to have a loss of ecosystem services. Can you imagine not having a picnic under a beautiful tree? And we'd have loss in terms of wood quality and wood products. And this also leads to a loss of carbon drawdown, which we really need our forests for. So if you look at records over the past 30 years, massive tree mortality is increasing. And the reports indicate that it is both biotic and abiotic stresses that are the cause. So what we really need are resilient trees. Resilience is the capacity to recover from such difficulties. Now, in South Africa, where I'm from, we rely on both eucalyptus and pine forestry. Eucalyptus is native to Australia and the surrounding islands, and it makes up 50% of the plantations in South Africa. In most cases, it is hybridized with Eucalyptus grandis. The pine species that we are using are actually native to Mexico and Central America, and we are using hybrids for improved performance. So, for example, hybridization for frost tolerance. So, what my team and I are really studying is how a tree is talking to a pathogen and how the pathogen is talking back. So to really understand this molecular dialogue, we use transcriptomics. Transcriptomics, as was described very nicely by Steve, is that we are looking at the message that is sent. So this message is then translated into the chemicals that a tree produces, and this is a branch of study called metabolomics. Now, really, this is all spearheaded by the production of the poplar genome, and thank you and congratulations, Gerald Tuskan. Soon after that, it inspired the genome sequencing of Eucalyptus grandis, and so we had a high-quality genome from which to derive much more information. So today I'm going to tell you about a curious canker that affects our eucalyptus trees, a brutal blight that affects young eucalyptus leaves, and then a nursery wilt disease that affects pine production. So this curious canker is caused by a fungus that is known as Chrysoporty austroafricana. And I share this with you because it is a relative of the pathogen that causes chestnut blight. This has led to the fall of the American chestnut. So in this picture, you can see some of the trees have cankers. These cankers render the trees quite susceptible to wind breakage. We have these controlled inoculation systems. So back in our laboratories, we've got these kinds of experiments that go on. We can apply the fungus in a controlled way, and then we can look for those that are resistant and those trees that are susceptible, and we can do some comparisons. So plants make hormones, and what we found is that in the resistant tree, there was a higher level of this hormone salicylic acid. So you would know salicylic acid is the main ingredient in aspirin, and it's almost like the resistant tree knew that a headache was coming, which was the fungus. It also had lower levels of jasmonic acid. So we also looked into putting this as a model of the dialogue, and what we identified was a difference in this gibberellic acid signaling. A particular gene is being downregulated in the resistant host at 3 days, while in the susceptible one, this gene is being upregulated. And so we tried to figure out what is happening and what is the fungus doing in the tree. The fungus itself is actually making this gibberellic acid, and it is promoting susceptibility. The resistant tree is able to stop this in its tracks. So we are first working on individual clones. And so we started moving into these structured populations. And here we had a family of a hybrid that was then replicated for us. So we worked with 2 industries back home in South Africa, and they've produced for us these replications that allow us to have about 12,000 trees. What this means is that we can go in and do in-field inoculations, and after a couple of months, we go back, and this is a type of cankers that form, and we measure the lesions that are produced. So we've been working towards this type of systems genetics approach, just looking first at our nursery type inoculations, our field inoculations, gathering all of the phenotypes, and then associating those phenotypic differences with the genetic differences. And the way we're doing this is we're using the 72,000 eucalyptus SNP chip that Dario mentioned earlier. But not only are we looking at the genetic level, we're also looking at particular metabolites that may be produced. And we're working with a colleague, Professor Almond Hammerbacher, to look at certain chemicals that may differentiate resistant and susceptible trees. Because, of course, it's very difficult to go in and work with 12,000 trees and go in and do those inoculations. Next, I'll tell you about this brutal blight. It's caused by a pathogen known as Teratosphera destructans, and in this picture, you can see the devastation it causes. The clone that is shown in the front of the picture is highly susceptible, and there in the background you have a more resistant clone. This pathogen affects those trees that are grown in tropical and subtropical areas, and also has become a problem in the nursery. This is the distribution of the pathogen, and when it was identified in South Africa in 2015, the first thing my colleagues and I had to do was to really put together a protocol to do these repeat inoculations to mimic the kind of conditions to get the best symptoms. In that way, we developed a scoring system, so all the way from asymptomatic to leaf deformation. This way, we could then screen plants and know whether they are highly resistant up to highly susceptible. So again, we looked at the transcriptome and how these plants were responding. The resistant plants very early on seemed to recognize the fungus, activating the correct type of defense responses. Interestingly, the susceptible hosts were only activating these kinds of recognition signals a lot later. So we were very curious about this early line of defense, and what we recognized was that on the leaf surface, where you had the fungus growing, around those areas, you also had leaf wax that was degrading. So we looked a little bit closer at the leaf wax composition, and in one of the clones, we found that after inoculation with the fungus, there was a particular compound called cyclooctanol that was increasing. And so we applied the cyclooctanol, and we applied this first in a Petri dish, and we saw that it has an effect on pathogen germination, where it is suppressing pathogen germination. We also applied this compound onto the leaf surface, and we found that increasing the amount of the compound decreased the pathogen germination. To the point that if we treated a plant that was considered initially highly susceptible with cyclooctanol, we then would find that it became more resistant. So we were able to change its particular ranking. I'll move on to talk to you about this nursery wilt, and it's caused by a pathogen known as Fusarium cercinatum. In the nursery, it holds production of susceptible species to almost 50%. And in mature trees, you can find this kind of symptom where you have this pitch that is oozing from the tree. And out on the landscape, if you see this kind of red flagging on a pine, it is typical of the symptom caused by this pathogen. It is still prevalent in many countries around the world. And our big challenge was to identify what underlies resistance against this pathogen. And so we again had a controlled inoculation system where we are able to clip the apical bud or the tops of the little pine trees, and we could apply the fungus, and we could do comparisons with treated with the fungus and not treated with the fungus. And we had the experiment where we had RNA extractions done at 3 days, 7 days, and we looked at both a resistant and a susceptible species. So Pinus technomanii is resistant to this fungus, and Pinus patula is highly susceptible. In the model, looking at the resistant interaction, what we saw that was really interesting is that at 3 days and 7 days, there is a type of consistent response. If you look at our susceptible interaction, very early on, you see that there's very little types of responses going on. But there's also, at 7 days, a terpenoid biosynthesis, which is quite an important part of defense, is actually being suppressed in the susceptible interaction. But it was really looking at what the fungus may be doing that got us to understand how the resistant tree is winning. We looked at the pathway that relates to agostral biosynthesis. So the agostral biosynthesis is important for the fungal cell wall integrity. And what we saw was that the resistant tree is actually inhibiting the fungal lipid biosynthesis. So from within the tree, there's suppression and compromise of the pathogen. And that is how we think that the resistant tree is winning. So we have been working towards this population where we are now examining using a precision phenotyping, where we are looking specifically at fungal biomass. So if we can count how much of fungus is present, we'll be able to get a better sense of resistance or susceptibility. We've also been working with field inoculations, and we also have a pine snip chip. So we have a tropical pine snip chip, and with that, we are now relating these phenotypes that we are collecting to the genetics. And so we have regions on the genome that we can hone in on that will be associated with resistance. So I've told you how we've been looking at this molecular dialogue, but the big question is, how do we begin to integrate these layers of information? And over the years, my team and I have studied various interactions with eucalyptus and different pests and pathogens. And what we're looking to do is now identify these so-called hub genes, that is central points that are being affected by different pests and pathogens. So we are layering information thanks to genome sequencing, the transcriptomics, proteomics, metabolomics, and even looking into the microbiome. And what we came up with is that central to all of these different interactions is that there's nitrate-associated genes. And this is going to be the subject of the next phase of our work, looking at functional genetic testing of selected candidates, doing some of the kind of work together with CJ to look at poplar knockouts and overexpression, and we can do some work in eucalyptus as well. So ladies and gentlemen, I've told you a little bit about the biology of resistance. It's quite complex, but we do look forward to using the outputs for DNA markers for resistance. And as Dario pointed out as well, genomic selection models will help us to do predictions for disease resistance. And we hope to achieve a tool for yield protection so that we have sustainable forests. I want to thank my lab back home. These are the people that make the work happen. And thank you to the funders that enable us to do the work. Thank you for your attention.
Anushka, thank you so much. I think we can all see this progression that I was talking about in the beginning. From 40 years ago, we move ahead. Next presentation, Catherine Bastien from INRA in France. Catherine is a quantitative geneticist, and she will share with us some information on genomic information to improve the quality of trees.
Thank you.
Catherine.
I would like first to thank you so much, Jerry, because you were the first who switched on the light on the first tree genome, and you will give us also the way to switch on the light of thousands of trees everywhere in the world on different species. And thanks to these lights, we are really doing transformative change in breeding. So thank you so much. I would like in my talk just cover some context points of poplar breeding, then go to why genome information is of interest in breeder toolbox, then give you some transformative changes occurred in poplar breeding the last 20 years, and of course, because we have young scientists in the room, to discuss about expected further research efforts. Poplar breeding. Poplars cover about 62 million of hectares in the world. That represents 63% of fast-growing tree area, and this fast-growing tree area is expected of major use for our society. Genetic improvement was, is, and still be the most adapted strategy to meet all demands for both adaptive, quantitative, and qualitative performance. And why? Because vegetative propagation is easy, and we can really take value of individual variation, because clonal forestry has been adopted for decades, so society has been used to that. And then, because clonal selection provides high genetic gains and rapid deployment, the International Register of Clonal Cultivars is more than 4,000 entries, with a lot of interspecific hybrid, and the process is continuing with more than 30 new entries every year. The current challenges for poplar breeding is really to remain poplar plantation and poplar natural forest, be able to maintain carbon storage sequestration. We need both use of forest. We need wood resources and the diversity of wood properties provided by poplar. Of course, Populus with its large genus is part of the biodiversity, and we need to keep it. And the last, we need also poplar to ensure land restoration, land protection. In the way of doing poplar breeding, it seems that, the breeding target change around. At the beginning, we talk about adaptation, but short adaptation. And in phases was given to growth, wood quality. But the carbon economy is more complex than single trait. Carbon economy, it's the link between all these traits. And adaptation in the context of climate change is increasing in importance, expected, increased resilience to abiotic and biotic risks. Regarding the genome information in interest for tree breeder, first, we start from genetic resources. And starting with a dozen of markers and now be able to use millions of markers change our lives. Regarding evaluation of performance, now we need to share new knowledge on the genetic basis of complex traits such as vulnerabilities and adaptive potential. That will help phenotypic prediction. Then, after evaluation, we have to process to selection and recombination, the cycle for breeder, and we need to adjust methods to accelerate and precise both selection and recombination. And at the last, it's not finished when you're a tree breeder. When tree breeder has to go into the field to see deployment. Deployment is the start of the use of this material. And we here see that we still need to use this information. Regarding genetic resources, the first most important information is that genomic information increase our knowledge of natural distribution on all different species. And population phenotypes are now known to be well predicted by genomic data as well as climate and geography. So that's something quite important for us to have this past history with us in the process of breeding. Regarding genetic resources, the breeder also is careful about relatedness on the material deployed, and so we have to compare. And you have on the left part, the comparison of the distribution of the Populus nigra genitors in different European poplar breeding program in comparison to the natural variation. And you can see that, hopefully, in Europe, we succeed with complementary breeding program to cover and be used of all this diversity. On the right side is just to show you that, of course, a breeder is also providing some varieties for restoration of natural population and landscape. And thanks to this genomic information, we were able to build 2 multi-clonal varieties, for example, to be used for river basin restoration in France, in Rhône, and these two multiclonal varieties are representative of a natural diversity in this population. Regarding evaluation of performance, first of all, we have this first result regarding QTLs in large mapping pedigrees. And when geneticists say that what was important is to detect QTLs, breeders are not at the same way to interpret the result. They learn how to describe better their genitors. It's not really QTL informed, but the way for the first time they identified what are the potential of their genitors thanks to these genitors of large families. And the second one is to be able to explore the Mendelian sampling effects, because when you have full SIB, theory gives you the same level of relatedness between all full SIB, and thanks to genomic information, you explore much more. Regarding evaluation of performance, then we have this huge effort of combination of genome polymorphisms and even gene expression studies to detect genomic variant in large population through GWAS. And what's important in GWAS for breeders also, it's this capacity to detect adaptive signature in tree genome. Adaptive signature is the imprint of past natural selection, and tree breeders dream to be as efficient as natural selection. So to learn how signature is distributed could help breeders already to also have signature on the genomes. Here you have the results, show you signature on Charcot isomerase gene located on chromosome 10, and it's linked to contour radial growth in Populus nigra. And you see here the signature of history in the eastern part of Europe on this signature. Last point, when you have a large genetic variation to explore, sometimes it's in the rare events where you find very huge interest. And identifying rare individual, rare alleles is really difficult without this genomic information. So here you have an example of the detection of rare recessive alleles. So due to the nice collaboration with WOW teams. And it's just to tell you that we were then able to just catch something interesting with a low allele frequency in natural population, but most also a low frequency as individual, because homozygous genotype are still also very rare. So here it's to show you the power of this genomic information to really detect rich information. Regarding selection, individual sequencing and high-density genotyping really paved the way for genomic selection also in popular. And what are the expected benefits? Of course, time gain, with precision gain. What's it? I think as scientists, we need to be precise. Better management of genetic diversity, gain in selection intensity, but also maybe save some money to maybe invest more in diverse phenotyping. It's a way to balance and to change. Regarding the results, here it's to show you that a proof of concept of efficiency of genomic selection, combining information on genome polymorphism and transcriptome in our Populus nigra breeding population. And you can see here that for 3 traits, height, circumference, and rust resistance, we have really multi-trait prediction efficiency. With the same set of SNPs, you predict many traits. We are not limited, so multi-trait selection is really the most important focus. Regarding the use of molecular markers, the SNPs from the genome, but also the transcript, show a better and higher level of prediction. And our breeding duration cycle was divided by 3. So genotyping costs now are not limited extra cost compared to phenotyping or archive cost. We really convince breeders, thanks to these other kind of course. The comparison, it's not to analyze the genotypic cost per se. Then, regarding deployment, just to say that it's important to facilitate traceability of natural and improved genetic resources at landscape scale and across country. Not only, all these resources are international resources. And of course, help the monitoring of gene flows between natural and cultivated stand. And this is a huge question, mainly raised by the society, gene flow. And surprisingly, in France, where people were afraid about this poplar plantation with exotic poplar species, they were not aware that, in fact, Populus nigra italica, which is from the same species of the natural population, in fact, is a more invasive with a high rate of introgression. So when you explain that to the society that ornament is more dangerous than plantation, you stop any discussion. So further transformation are still needed to progress, to meet the challenge. We need new knowledge. Really, we need to identify and characterize rare genetic diversity to preserve it. It's time, even maybe it's too late. Genetic and epigenetic basis of plastic response. Plastic response is really to have a better view of short response because we have a very heterogeneous climate and it's important to have this short response. To identify genetic limits in key function response, and that the links with, these biological understanding and the role of functioning. And these limits, we must know them in extreme environment, and we need to know the limits regarding the allelic composition. Genomic options also have to resolve trade-offs because multi-trace selection is not so easy. We have a lot of trade-offs. Regarding transformation, we need open new tool. Open is important. We can see that we start in this integrative modeling. It's increasing the last few years, and we have to continue to do this more modeling work. To do that efficiently, we need open databases. Open databases to collect all information, genotypic, transcriptomic, epigenomics, phenotype at different level. And we really need to have this multi-source information connected. Then, and the last, for me, it's important that we continue to train and that to train differently tree breeding and genomics, and in order to update skills in genomics and breedings, and also to be able to integrate new jobs in our breeding sector to integrate this genomic technology. Thank you so much. I want to thank Jerry, all the actors of the international poplar breeding community, the Marcus Wallenberg Foundation to give me the possibility to be here to exchange with you. And thanks also all national, private and public funding institutions, and to thank the European Commission for their precious financial support to develop more efficient forestry breeding. We need them to continue to have lights.
Thank you so much, Catherine. A wonderful presentation and a beautiful picture. And yesterday, in a little run through, you also mentioned the technicians in the labs.
Yes.
Which you didn't now. But I think all of us that work in the field of science need to remember them too. They are so incredibly important. We're going to move on. We have been, we're going back home. SLU, yes, Harry, please. Harry Wu, we started in America and we've been through Brazil and Africa and we've come back to Sweden. Harry is from SLU and focuses on conifers, which is a little bit of a change, which is really good. Welcome, Harry. Thank you very much.
Thank you very much, Mike, for the introductions. So now I will talk about this genomic breedings in the Nordic conifers right here in Sweden, the home countries of this Wallenberg award. So, and as many of you know, we have two big conifer species in the Nordic countries, Northern Europe. First one is the snowy spruce. So that's distributed mainly in Northern Europe. In Sweden, we have a large planting, about 250 to 300 million seedlings. So that's a very important commercial species. So second one is the Scots pines. This is the most widely distributed conifers in the world. Also important, in Sweden, we plant about 150 to 200 million seedlings each year. So these two main conifer species support our industries in Sweden. So, but these two species now has been breeding for about 70 or so years, but we only had 2 generations for Norway spruce and 3 generations for Scots pine. The major concern, the constraints of such and low generation turnover is long-term progeny testing, not only for us, also many conifers. Also for some conifers, they will flower very late. So there's 2 issues we need to resolve. With recently genomic breeding come up last about 10 or 15 years, so we saw the selection on DNA marker is an obvious choice. As Dario indicated, we can select at the seedling age. So if we can mate them, then we can shorten the breeding cycle from about 30 years now for Norway spruce to about 13 and maybe 15 years. So at least we can have double the generation if we can do the selection and the early flowerings for like Norway spruce. For Scots pines, it's better because they flower earlier than the Norway spruce. The genomics breedings starts with reference genome, if it's possible. So we build this shotgun, this genome for Norway spruce about 10 years ago, but that's very fragmented using shotguns. So the reason is we have 20G genome for spruce and 24G for pines. So that's about 50 times and 6 times of size of the Jerry's cottonwood. That's why we are very much behind than you. So, but we were able, last few years, we were able to assemble those 2 species and with a group of people. Then those are PI of this project, but we have many people involved. So what we found is even though those 2 species separated about 130 million years ago, so their genome have remarkable macrocentennies. So all 12 chromosomes, many genes, and they correspond among those 2 species. So, and the main find is genetic innovation is mainly by novel gene copy creation after their separations with novel function or differentiated functions. So now after reference genome, we need to do genomic selection in the genomic breedings. So with genomic selection, we need genotyping tools. Initially, we developed the exome sequence and the GBS, but the last 5 or 6 years, we mainly focus on using this SNP array. So we developed this 50K SNP array for Norway spruce, for Scots pine, using this new genomes. So we mapped all this 50K to the Norway spruce genome and this Scots pine genome. They are more or less evenly distributed among 12 chromosomes. So as Dario indicated, so this now the genomic selections. So we have this genomic tool, so we can genotype many trees for genomic selections. But the principle and approach has been indicated by Dario, so I will not go through it. So mainly, and we did 6 studies of genomic selection in Norway spruce. Mainly we're using this selection accuracy between genome-based methods, between phenotypic methods. We compare how effective using genomic selection compared to the phenotypic-based selections. So here I give 2 studies. We're using, first one, we studied the growth traits of tree height, dBH, and the water quality traits of water density, microfibril angle, and MOE. Second study, we also studied similar growth and the water quality traits, plus phenology traits, like bud flushing time, frost damage. So from all our 6 studies, and I didn't present here, so here is similar. We found the efficiency, selection accuracy from genotype, almost the same as using phenotype. So we're using this relative efficiency from genotype to phenotype as the benchmark to assess genomic selection efficiency. So with this similar efficiency, that means if we can select at a seedling early age, shortening breeding cycle half, we can almost double genetic gain per year. So our study actually very similar to all published conifer genomic selection across using cross-validation of some generations. So even though this relative efficiency between genotype selection and phenotype, they are varied among different species, among different traits, but all together, actually, they are almost one, similar, 0.97, I summarized. So that means genomic selection almost the same effective for many, many different traits and for conifers. So this is all well. That means now we come to go into genomic selection. The one issue is actually concern us apply to the industries. Because and the breeders' main concern the cross-generation genomic selection efficiency. Because we were able to build a model from parental generation, we usually make cross-validation on the same generations. They are almost same effective with selection with phenotype. But for us, we usually make strong selections. Then we only select like 1%, maybe 2% best trees, make a cross, produce the progeny. So we only have a phenotype, build a model from parental generations. So whether that model will be still effective for selection at progeny, because we have a strong selection effects. From quantitative genetic theory with strong selection effects, actually the allele effects will change. So also there's mating effects. So we want to see whether it's still okay for our breeding purpose. So we did a very large studies using 2 generation materials. We have using parental generations and about 1,000 clones tested on about 13 sites. So then we selected the best 30 clones, make a cross, make a G1 generations. So those are tested on 3 sites. Then we build, we genotyped about 1,000 clone parental generation and 1,000 clones for progeny generation. We build a model for each generation, have a cross-validation, then calculate efficiency with phenotype. Then we also use a parental model to predict the progeny genomic selection efficiency. What do we find within 7 generations? Their genomic selection efficiency is very high, whether use all 1,000 clones or just using parental clones with their relatives. So it's a different way. But we're using these models from parents generation, predict progeny, their cross-generation prediction, their efficiency dropped about 60%. So that means our selection, strong selection, have big effects on the effectiveness of genomic selections. Second, like in Norway spruce, we are very interested in this clonal selection within family for deployment. So we tested using global selection model from population for within family selections. So actually they dropped more, about 70% drop for within family rankings. So there's issues there for cross-generation for within family selections. So we also trying to confirm whether this is in another set of real data. We did this individual family clonal selections using 6 largest families. We built a model, global model for breeding selection. They are very effective genomic selection, even more than using phenotypic selections. But for individual family clonal selection, so their effectiveness about dropped to 47% relative to global selections. So we thought about why that's the case. Because they are in the first generation, second generation material, they have actually very, very low LD. So cross-generation is not effective. So we need some strategy to overcome such low effectiveness. So then we did simulations because we say we don't have many genetic materials. So we did a simulation. We set up 35,000 SNPs across Norway spruce genome with 500 QTL distributed. So what do we observed? 2 generations similar. This is the within generation, high effective. Cross generation, they dropped, but they dropped only 40% less than the real data. Similarly, for within family selection, dropped about 35%, dropped less because also it's also dropped. So from real data simulation, so we have issue for cross-generation genomic selection for within family selections. So then we did say, because now we have most conifers have 3 and 4 generations of breeding program. So we thought about it, if we're using more generations, more different generations, with the build of LD, whether that will overcome this issue, low cross-generation issues. So we did a simulation using 5 generations. We're using 1 generation model for next generation selection. We're using data from several generations together, all previous generation build model for next generation selection. What we found is as generation advanced, the genomic selection cross-generation increased. That's because of build-up of LD. So after 2 or 3 generations, actually their accuracy almost similar, only a little bit drop compared to phenotype. So now we are confident with advanced tree breeding program, we can implement both breeding selection and within family selection, particularly when using all the data previous build model for such genomic selections. So now we apply this genomic selection to 18 Norway spruce breeding population across Sweden. So we genotyped about 10,000 trees so far. So we have built a model for all those generations. However, this the genomic selection is only half story of genomic breedings. We need to have those trees have early flowerings. So now we started this big project, the rapid cycle breeding, under the leadership of Uwe, to combine genomic selection and gene editing for early flowerings. And Yen and Uwe, they discovered a single mutation at this microRNA 156 binding sites of Norway gene SPL7s. Then they can activate this early flowerings. So we're trying to gene editing for breeding trees to have early flowerings. So now we have the genomic selection. We have the model for selection at the SCE levels. We're using the seeds, develop this somatic embryogenesis. Then with that, so we make gene editings and introduce this SNP. It's called the eclo SNP. Then tree can flowering 3-5 years instead of 20 years. So after flowering, we're mating with the other elite breeding trees. Then they separate 50% eclo SNP clones can be separated. So another 50% we can use it for deployment for genomic selection. So overcome the regulation issues with gene editings. So we also did this genomic breeding for Scots pines. We had 3 studies. We had similar using genotype, almost 90% effective compared to the phenotype. So now we apply this genomic selection to Scots pine in 7 breeding populations and in Sweden. In Scots pine, we have 3 generation breeding material. So we are using build model using 3 generations. That will be more effective for selection for next generations. So I just have a very simple summaries. So genomic selection at the early seedling stage for breeding and the clonal selection works for conifer if long LD linkage disequilibrium builds up through advanced generation and multiple generation breedings. But early seedling genomic selection for some conifers must be accompanied by early flowering using genomic tools, such as gene editing. So we are combining quantitative genomics with this biotechnology tools. So I try to bridge the gauge between, Steve said, between this genomics breeding with this biotechnology. This is the acknowledgement for the team and for the collaboration and for the fundings. So I will mention here, there's many funding, mainly Wallenberg Foundation. They funded us several big projects, including this genome, reference genome, resequence, and genomic selection. Thank you.
Harry, thank you. Thank you very much.
Thank you.
I had to smile at Harry making the point about pines or conifers are difficult, long, and eucalyptus and poplar much shorter and easier. But I often say to my colleagues that work in the agronomic field, I wished I worked on peas or beans. Isolines, three generations a year. That's what we really need. Yeah. Thank you so much. We need to come to an end.
Yeah. Thank you.
You go that way. So, to wrap up and not take too long doing so, because there is something special waiting, and some of you know this and some of you don't. And these things have changed here, and that is that we get to eat the Wallenberger. Yes? Is it still there? I think this is a long-standing tradition, very, very special, something to remember. We come to the end of an amazing symposium. It's been truly inspirational, and I keep thinking back to that starting point, Jerry. 20 years ago, and it feels like just yesterday. And then you've got to add on another, how many, 5 years before the paper came out in 2006. Just incredible, but got all of this started like a huge knock-on effect. Like, it's bigger than a wave. It's like a tidal wave, isn't it? It's just absolutely amazing what has happened from that first point of first plant genome using shotgun sequencing, just that idea and maybe the Human Genome Project kind of got that going, thinking again about what's happening in the medical human field, to so many discoveries. And I started making a list. From the talks and background information also, and it just goes on and on and on. You didn't mention here, but I think last night you did, Jerry, discovering the first microbiome in a tree. I read that. I read the paper. I didn't go into the background. I didn't know that, actually, and I work in the world of microbiology deeply. So that discovery and what's come out of that tree breeding, approaches to tree breeding, the technologies, these growing technologies, and we've heard many of them shared with us today, the discovery of genes, and that intersection of new gene discovery, so incredibly important. Think about those, the ant bacterium genetic material in the trees, and what that's all going to mean in the future. We're learning how trees grow. So those genetically modified or edited trees, CJ, with more leaves, more growth, drought tolerance, disease tolerance, photosynthesis. I mean, this is just unbelievable. We're at the cusp. We're just at the cusp of something bigger, much, much bigger. So those of you, the young researchers here, just such an incredibly exciting time to be working in the field of science. I realize you're not all in the working in the field of genomics, but it's all the same thing. So I've been thinking about what is the missing link? Where is, other than the issue of dealing with regulation and the perception of people, where could we do a lot better? And I think it probably lies, at least in my mind, and it's my own perception. You can disagree with me over the Wallenberger, but something about the intersections between disciplines. To really build on those intersections, remote sensing. We heard a little about that's coming. Remote sensing, be able to, from your phenotyping in a laboratory or a greenhouse environment into the whole tree, the individual tree. So to get the cross linkage between all of these important disciplines. It's engineering, it's social science, very important. Social sciences, molecular genetics. So that I think is the space. So for those of you that are younger here, you wouldn't probably have read a book, which it's one of my favorite, and I'd like you to go and read it. And I'll come and ask you, maybe I'll write to you. And the book is written by Frans Johansson, and it must be about 15 years old. How many people have read this book? Please read it. It's a little book, and it's called the Medici Effect. And the subtitle is something like this, and I don't remember exactly. If you read this little book and you don't come up with 2 Mona Lisas, you're not thinking. It's fabulous. It's small. It's a few hours, maybe a morning of reading. Read that book, and I think that is that whole issue of the intersections between disciplines that is really going to define the big future. We've had a wonderful morning. We've sat for a long time. It's been a delight for me to be here with you. Thank you for listening, enjoying the talks, to wonderful speakers. Dario, hopefully still online. Thank you for patience, for keeping to time, for making this all a lot of fun. I really, really appreciate that. We all appreciate it. There's a lot that lies ahead. It's a lot that's very exciting, but now it's time to have lunch and to wish you all safe travels home, and we see you again soon somewhere in the world of science. Thank you so much.